Good morning, guys. Hope you all are fine and have been preparing very well for your upcoming exam. In this hard efforts of yours that you guys have been putting in so far, we on behalf of MIST are also here coming to help you one more time with the sessions of revision video. And in this revision video, although you guys have put lot in of hard works, lot of efforts in writing those workbooks during those long 12 hours of classes day in day out, we really appreciate your efforts. Now in order to take things further so that you guys can remember the main crux of all the subjects that will be helpful for you in MCQ solving on the D day, please try to understand. we shall be talking about we shall be talking about these revision sessions of biochemistry as well, that will help you in focusing on the main most highest yielding topics or the concepts from biochemistry. So here we are meeting one more time, my dear friends, although not face to face but in the online mode. We are talking about this revision video, so let us just enjoy this journey and try to keep these sessions and try to treat this session also as chanting sessions, my dear friends, so that whenever we are going through this, you also please try to remember and retain the main points that we are going to discuss one more time. Here the content will be very limited, not as big as like what we have been writing in the workbook sections, but here we are focusing mainly on the most important points. So this will be mainly like an quick rapid fire section from biochemistry. We will be going chapter wise, and all these points, my dear friends, you have already written in your workbook, so we are just quickly recapitulating all those, right? Now please try to understand first thing first, my dear friends. Here we will be marking the most important points that we have been discussing so far. Now please remember, I'll be using one yellow common highlighter over here to focus on the most important points. Yes, so without wasting much time, let us quickly talk about this. Enzymes. We have seen that there was something called as denaturation of proteins, my dear friends. Denaturation was the primary structure of proteins remaining intact, however the secondary, tertiary and quaternary structure getting lost. That was denaturation. Important point, try to be remembering. Ribozymes, we have seen that these are the RNA having enzymatic activity. RNA having enzymatic activity, and we have seen the name of that RNA, that is peptidyl transferase, that is needed for our peptide bond formation, right? Important point. Mode of action of enzyme: decrease the activation energy of any biochemical reaction. Yes, coming to the monooxygenase, important point. They are also known as hydroxylase or mixed function oxidase. Yes, we have seen hydroxylase are those enzymes where we have formation of the hydroxyl group and the two oxygen were performing two different jobs. That's why mixed function oxidase. Let us quickly see this important point. Nitric oxide synthase, NOS, is a dioxygenase. We have seen this. Kinase, we have seen, belongs to transferase. They adds phosphate. Whereas if we talk about phosphatases, my dear friends, they remove phosphate. Yes, important technical points. Coming to blotting techniques. Blotting techniques, we have seen, my dear friends, mnemonic: snowdrop, north mein roti, south mein dosa, west mein pizza, as well as east mein egg. Please try to understand important points. Southern blotting, this chart is important. Southern blotting for DNA, northern for RNA, mainly mRNA, western blotting for proteins, and eastern blotting, latest question, post translational modification of proteins, that is also for detection of enzymes. East mein egg, please try to understand this. Sandwich ELISA, we have seen, sandwich ELISA primarily detects antibody. Important point. Holoenzyme, not hollow, holoenzyme, two part, apoenzyme plus coenzyme part, protein plus non-protein prosthetic part, holoenzyme. Previous year's question. Let us try to target important points. Please remember, mode of action of coenzyme accept one of the product of the reaction or one of the product of the reaction by reactions. Please try to remember the chart that we have seen for coenzymes active forms. Active forms B1 TPP, B5 CoA, B6 PLP, B9 tetrahydrofolate. These 4 questions were asked. You have already marked in your workbook, my dear friends. B1, we have most important function oxidative decarboxylation. B6 non-oxidative decarboxylation. B7 carboxylation, and vitamin K, my dear friends, gamma carboxylation. Yes, we have seen these 4 important point. B5, please today's class try to focus only on the points that I'm marking as yellow highlighter. B5 acyl group carrier, B6 also both transamination and oxidative deamination. And we have seen even in carbohydrate, glycogen phosphorylase stores 80% of B6 as pyridoxal phosphate. B9 one carbon pool, one carbon compound except CO2, B12 transmethylation reaction, vitamin C hydroxylation, right? Important points to be noted, my dear friends. And this gamma carboxylation was dependent on vitamin K, where we have the clotting factor 2, 7, 9, 10 are getting modified from glutamate to gamma carboxyglutamate because those GLA residues were responsible for calcium chelation. Coming to zinc containing enzymes, mnemonic was super carbon pagal. We have talked about this one. Please try to understand, my dear friends. Most important enzyme we are targeting SOD, carbonic anhydrase, ALP, ADA, and dehydrogenases. Important point to be noted. Zinc containing enzymes. Now we have also seen the importance of alkaline phosphatase, my dear friends. Alkaline phosphatase. Please remember, we have seen that drastically high levels of alkaline phosphatase, we have to think about bone disease. Try to mark the most important point, my dear friends. Drastically high levels, bone disease. Drastically high level bone disease, 10 to 12, 25 times than the upper normal limit. Normal limit was 80 to 125 international units per liter. Very high levels of ALP, think about extrahepatic obstruction, obstructive jaundice. 4 important parameters for diagnosing obstructive jaundice: conjugated bilirubin 50% or more, then alkaline phosphatase level very high, 4 to 12 times, up to 10 to 12 times we had written. Please try to understand. Also we have raised prothrombin time and raised GGT. Raised prothrombin time and raised GGT. Important point to be noted, my dear friends. Most common cause in India is gallstones. Moderate or slightly high. This is 2 to 3 times. Think about moderate hepatic disease. Important point to be noted. Try to understand, my dear friends. Please remember this. ALP high, GGT normal, think about bone. ALP and GGT both raised together, think about obstructive jaundice. And ALP normal, isolated rise of GGT, think about alcoholic liver disease. Right? Isolated GGT, alcoholic liver disease. Important point to be noted. Coming to ADA, adenosine deaminase deficiency, leads to SCID, deficiency SCID. First successful gene therapy, my dear friends. We have talked about. Try to understand this, my dear friends. Important point to be noted. CSF ADA more than 8 to 10. TB meningitis. Important point. Ascitic fluid ADA more than 36. TB peritonitis. Copper containing enzyme, please remember the mnemonic PCM salted, right? Important point to be noted. PCM salted, important point. We have most important cytochrome oxidase ceruloplasmin, MAO, SOD, ALA synthase, rate limiting enzyme of heme synthesis, lysyl oxidase, tyrosinase, and dopamine beta hydroxylase. CM salt, at least remember CM salt. That has been already asked in your exam. CM salt. Ceruloplasmin, my dear friends, it is an enzyme having two enzymatic activity. First one, peroxidase, ferrous to ferric in the blood so that it can be carried by transferrin. Also acts as copper binding ATPase that is present in the liver, which is needed for the copper excretion from liver to bile. Yes, Wilson's disease due to deficiency of the ceruloplasmin due to the ATP7B gene mutation. ATP7B gene mutation. Deficiency of ceruloplasmin, my dear friends. Copper binding ATPase activity is lost. So liver will be now storing more of copper. We will be first having hepatocellular degeneration. Then we may have later on cirrhosis of liver as well as lenticular degeneration of the basal ganglia and green or golden colored pigmented rings over the cornea, around the cornea. We have KF ring, Kerr-Fleischer's ring. And ultimately over the lens we have chalcosis that will cause sunflower cataract. Chalcosis, any foreign body containing copper. Try to remember them. Important point. Cytosolic SOD, remember copper and zinc dependent, while mitochondrial SOD is dependent on manganese. Previous question these are. Both of them has been asked. Do not miss this. SOD gene mutation, remember Lou Gehrig's disease or amyotrophic lateral sclerosis, may be remembered. Important point to be noted, my dear friends. Then we have talked about selenium containing enzymes. Please remember there are also selenocysteine containing enzymes, glutathione peroxidase, thioredoxin reductase and deiodinase. Peripheral conversion of T4 to T3 is done by D2 deiodinase, better answer than D1 deiodinase. Selenocysteine, my dear friends, 21st amino acid. It is derived from selenium and serine and also coded by the stop codon UGA. Glutathione peroxidase, it is important in RBC membrane integrity, prevents hemolysis. Peripheral conversion we have already seen, D2 deiodinase best answer, T4 to T3 peripherally, T4 to T3 deiodinase number 2. Xanthine oxidase, my dear friends, molybdenum containing. Apart from molybdenum, it was containing iron and FAD both. Important point. Coming to Km. Km is substrate concentration at half maximal velocity. Is try to understand, my dear friends. Important point to be noted. Km, and we have also seen only 50% enzymes are saturated at that particular substrate concentration. It is independent of enzyme concentration and it is also the signature of an enzyme. Right, important point to be noted. And Km high means affinity low. Km low means affinity high. So inversely proportional to affinity. Taq polymerase, thermus aquaticus polymerase. It is important in our PCR technique because it can withstand very high temperature for thermal cycling. PCR technique. PCR, my dear friends, it is dependent on also two ion, that is magnesium and zinc. Important point to be noted. PCR dependent on magnesium and zinc. Conventional PCR, it is basically for the amplification of DNA. RT-PCR amplification of mRNA. Important point to be noted. mRNA, please remember this. Right. Important point. Competitive inhibition, inhibitors block the substrate binding sites of the enzyme and are usually reversible on adding excess substrate, while non-competitive inhibition is irreversible. Competitive reversible by adding extra substrate, non-competitive irreversible, so substrate addition is useless. Important point from these three inhibition, please try to understand. Km and Vmax. Km increased, Vmax no change, competitive. Km no change, Vmax decreased, non-competitive. allosteric, Km increased, Vmax decreased. Uncompetitive bouncer question, Km and Vmax both decreased, uncompetitive. Enzymes that are common in active in phosphorylated state, only four. Glycogen phosphorylase, hormone sensitive lipase, ATP citrate lyase, and RNA polymerase 2. These are the four enzymes that are mainly active in phosphorylated state. Isoenzymes catalyze same reaction, important point. Isoenzymes catalyze same reaction in same or different tissues. Same reaction is the question, my dear friends. LDH isoenzyme, please try to understand, highlight the points. LDH1, please try to understand, flipped pattern, most abundant in heart. Flip pattern, 1 more than 2. Normally in the plasma 2 is more than 1, right? LDH3, lungs, so COVID-19 patients, LDH4, my dear friends, most abundant. If they ask you by chance this question, this we have not talked during our face-to-face classes. Most abundant, if they ask, most abundant in pancreas, but usually they ask you question on the skeletal muscle and the liver. So please remember, for 4 we can also remember skeletal muscle. LDH5, most abundant in liver and skeletal muscle. Answer to be marked as 5 more than 4. If they deliberately ask fourth one is most abundant where, please mark the answer as pancreas. But if they ask like this, most abundant in liver and skeletal muscle, then fifth one is a better answer than the fourth one, my dear friends. Please try to understand this. LDH isoenzymes, important. Fourth most abundant in pancreas, but in muscle and liver, fifth is more abundant than the fourth one, right? Important point to be noted, my dear friends. Coming to creatine kinase, please remember, CK1, brain ischemia. CK2, my dear friends, myocardial infarction, marker of choice for reinfarction. CK3, rhabdomyolysis, muscular dystrophies. Most abundant is the CK3. Most abundant. Marker enzyme glucose 6 phosphatase is microsome. LDH, cytoplasm, ATP synthase, inner membrane of mitochondria. 3 important questions that we have talked about. Coming to biological oxidation, ETC, connecting link of entire carbohydrate, protein and fat metabolism is acetyl CoA. It is acetyl CoA, my dear friends. Please try to understand. Connecting link of carbohydrate, protein and lipid, it is acetyl CoA. Please remember, immediate source of energy in the muscle up to first minute, answer is creatine phosphate. Up to first minute, answer is creatine phosphate. Up to first minute, rapid production of energy in the muscle. After first minute, my dear friends, answer to be changed as muscle glycogenolysis. After first minute, muscle glycogenolysis. Up to first minute, creatine phosphate. After first minute, muscle glycogenolysis. Important point to be noted, my dear friends. ATP is produced by oxidative phosphorylation in complex number 4, ATP synthase, FTP synthase. Beta subunit of F1 produces ATP. Which complex has no role in ATP production? Answer is complex 2. Complex 2 doesn't take part in ATP production. Important point to be noted, my dear friends. Please remember, oxygen is utilized in complex 4 and water is produced also in complex number 4, but ATP is synthesized in complex number 5. Important point to be noted, my dear friends. Please try to understand this. Right. Coming to the inhibitors of the electron transport chain and oxidative phosphorylation, please remember, complex 1 inhibitor, alka cheers, roten pears in bar. alkyl guanides, chlorpromazine, rotenone, that is also a fish poison, pyridine A, and barbiturate group of drugs. Alka, never forget Alka. You know who is Alka. Alka cheers, roten peers in bar. Important point to be noted. Complex 2 inhibitors, my dear friends. Carboxyl malonate. Carboxyl malonate. Malonate is also the inhibitor of succinate dehydrogenase of the Krebs cycle. Succinate dehydrogenase of the Krebs cycle. Complex 3 inhibitor, we have antimycin A and British anti-leucide. That was also an antidote of war gas, but nowadays no longer used. Complex 4 inhibitor, we have got 4 blockers. 4 blockers. Carbon monoxide, cyanide, azide, and hydrogen sulfide. Hydrogen sulfide. Cyanide, my dear friends, blocks. Cyanide, my dear friends, blocks cytochrome A3. A3. This one blocks cytochrome A3. Carbon monoxide displaces oxygen. Important point. Complex 5 inhibitor, my dear friends, oligomycin. Basically blocks flow of protons via FO base plate. FO base plate, right? Important point to be noted, my dear friends. Cyanide blocks cytochrome A3. Cyanide blocks cytochrome A3. Transport inhibitors, atractyloside and bongregate toxin, ADP in, ATP out blockers. Latest question, ADP in, ATP out blocker. 2,4-dinitrophenol uncoupler, physiological uncoupler, thermogenin, UCP1, mainly important one, non-shivering thermogenesis in children, thyroxine, bilirubin at high dose, essential fatty acid deficiency, long chain fatty acid excess. Most important one, thermogenin or UCP1, physiological uncoupler. Physiological uncoupler. Chemistry of carbohydrates. We all know what is Atkins diet. Diet to be prescribed for obesity: low carbohydrate, high protein, ketogenic diet. Low carbohydrate, high protein, ketogenic diet. How carbohydrate excess can be converted to fats? It is through overproduction of DHAP. Overproduction of DHAP. Exogenous carbohydrate is converted to endogenous fat in the liver. Ultimately, obesity and atherosclerosis. Please remember, glycoproteins formation is by glycosylation, whereas glycation process, please try to understand, my dear friends. Example is HbA1c, glycated hemoglobin. Glycated hemoglobin. Diagnosis of diabetes when value is 6.5% or more than that. Even this one, 6.5% or more than this is diabetes mellitus. Measured by the technique HPLC, high performance liquid chromatography. Main culprit for our diabetic complications, my dear friends, main compound of diabetic patients is this advanced glycation end products. AGEs. Ages comes with age. Ages comes with age. As the patient grows old, there is more chance of this age formation. What is age? Advanced glycation end products. Main culprit for diabetic complication. Please try to understand, my dear friends. Important point. Coming to proteoglycan, that is the carbohydrate part is known as glycosaminoglycans. Important point. High hyaluronic acid, please try to understand. It is most abundant in synovial fluid, vitreous humor of eye, no sulfate, no sulfate. Dermatan sulfate, please remember, blood vessel, hard valves. Important point, hard valves. Keratan sulfate, my dear friends, maintains corneal transparency. 2020 August FMG exam question. Most abundant in bone and only gag, no uronic acid. Only gag, no uronic acid. No uronic acid. Heparin, only intracellular gag. Heparan sulfate, basement membrane. Chondroitin sulfate, that is the most abundant one. Please remember, it is most abundant in cartilage, tendon, ligament, and aorta. Hard valves, please choose the answer as dermatan sulfate. Aorta, change your answer to chondroitin sulfate. Overall, most common is chondroitin sulfate. Right. Important point to be noted, my dear friends. Coming to those mucopolysaccharidosis, please remember Hurler syndrome, corneal clouding, autosomal recessive, Hunter syndrome, sensory neural hearing loss, X-linked recessive. Important points, please remember. X-linked recessive. Important point to be noted, my dear friends. Please remember. Enantiomers. Please remember penultimate carbon, fifth of glucose. D and L isomerism. D and L isomerism. Important point. Please remember them. D and L isomerism. Please remember. Glyceraldehyde, please try to understand, is called as reference carbohydrate or simplest carbohydrate. Apimers, please try to understand. Mannose and glucose C2, galactose and glucose C4 apimer. Important question to be known. Important point to be noted, my dear friends. Total number of isomers possible for glucose is 32. Total number 32, right? POCT measurement detects beta anomer of glucose only. Beta anomer of glucose only. Important point to be noted, my dear friends. Please try to understand. Sucrose, invert sugar. Sucrose, invert sugar. Lactose, beta 1,4 glycosidic linkage. Beta 1,4 glycosidic linkage. Lactose intolerance, lactase deficiency, diarrhea, vomiting, flatulence, abdominal cramps, child cries immediately after birth. Immediately after birth, abdominal cramps, immediately after birth. Benedict test for reducing sugar as well as reducing substance, semi-quantitative test. Semi-quantitative tests. Carbohydrate. Please remember, metabolism. Only fuel for survival of RBC is glucose. Only fuel is glucose. So end product of glycolysis in RBC is lactate, not pyruvate, because no mitochondria. No mitochondria. So all pyruvates will be finally converted by lactate dehydrogenase type 2 to form this lactate. Please try to understand. Cancer cells derive nutrition from anaerobic glycolysis, Warburg's hypothesis. Important point to be noted. Cancer cachexia, please try to understand. Mainly it is TNF alpha, but also lactate. Also lactate. Important point to be noted, my dear friends. Please remember, first committed step of glycolysis, PFK1. Please try to remember this. Glucose 6-phosphate is the connecting link of entire carbohydrate metabolism. Glucose 6-phosphate, golden MCQ points of glycolysis. SLP of glycolysis, please remember PK and PGK. PK and PGK. PK and PGK. Please try to understand. Incorporation of inorganic phosphate. Please remember glyceraldehyde 3-phosphate dehydrogenase. And this step also produces NADH plus H plus. Please try to understand 3 irreversible steps of glycolysis. First one, then the third one. Please try to understand. Then the third one, and try to understand. Also, we have talked about last step of glycolysis, phosphoenol pyruvate to pyruvate. 1, 3 and 10. 1, 3 and 10. First one, third one, last step of glycolysis. 1, 3 and 10. Please remember, insulin promotes glycolysis by regulating 3 main key enzymes of glycolysis, except which one? Except hexokinase. Yes, except hexokinase. Please try to understand, my dear friends. Insulin, mainly anabolic hormone. Insulin, mainly anabolic hormone that promotes glycogenesis, protein synthesis and lipid synthesis. Important point to be noted, and all these are seen in well-fed states, same as also like promote glycolysis also seen in well-fed state. Only reversible kinase of glycolysis, answer is phosphoglycerate kinase. Reversible kinase, phosphoglycerate kinase. enolase is blocked by fluoride ion. So additive used for blood sugar estimation, sodium fluoride and potassium oxalate. Sodium fluoride and potassium oxalate. Important point to be noted. Please remember, net gain, aerobic glycolysis 7 ATP, anaerobic condition 2 ATP. Important point to be noted. But if it is RBC, even in aerobic also it will be 2 ATP. 2 ATP. Right. Coming to the difference between hexokinase and glucokinase. We know there are 4 isoenzymes. Here we have 2, 1, 3, here we have 4. Please remember, sites. Hexokinase 2, if you say, then mainly it is important in our skeletal muscle. Skeletal muscle. But hexokinase 4 is mainly asked in your exams. Please remember, liver most important. Pancreas second, hypothalamus recently asked question. Hypothalamus recently asked question. Please try to understand, my dear friends. Activity wise, please try to understand. HK number 2, it is active at low glucose to meet the needs of brain cells, so that even if the sugar falls below 50 milligram per deciliter, the brain will still get the supply of glucose, because glycolysis in the muscle will be inhibited, right? And HK number 4 is activated minimum 100 milligram per deciliter, more than or at least equal to 100 milligram per deciliter, my dear friends. Please try to understand. So diabetes mellitus mainly dependent on HK number 4. Hemolytic anemia, PK deficiency, mechanism pump failure, key enzymes of gluconeogenesis bypass reactions. We have PC, pyruvate carboxylase, pep carboxykinase, and fructose 1,6-bisphosphatase and glucose 6-phosphatase. Please try to understand. Pyruvate carboxylase was the one that is present in mitochondria, and remaining reactions are mainly in the cytosol, but the last one is actually in microsome. So sequence we have seen is mito-cyto-micro, 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 mito-cyto-micro. Important point. Please remember, mnemonic PUBG. We have seen multiple times. PUBG, pyrimidine synthesis, urea cycle, heme synthesis, and gluconeogenesis. That is both in mitochondria and cytosol. Both in mitochondria and cytosol. Please remember, most important organ is liver. Most important organ is liver. Please remember, substrates for gluconeogenesis. Parle-G, important point. Parle-G, pyruvate propionate, alanine lactate, glycerol and glucogenic amino acids. Alanine contributes maximum. My dear friends. Propionate, principal glucogenic fatty acids. Question. Total 6 ATPs are required for gluconeogenesis. Total 6 ATPs are required for one molecule of glucose synthesis from 2 molecules of pyruvate or lactate. Important point to be noted, my dear friends. Glucose 6-phosphatase, it is absent in muscle. So no gluconeogenesis in muscle. Important point to be noted, my dear friends. No gluconeogenesis. PFK2 bifunctional enzyme. So please try to understand that fructose 2,6-bisphosphate is one allosteric activator of PFK1. Fructose 2,6-bisphosphate was the allosteric activator of PFK1 in well-fed state. Promote glycolysis, prevents gluconeogenesis. One hand help PFK1, another one blocks the rate limiting enzyme of gluconeogenesis, that is fructose 1,6-bisphosphate. Mallet shuttle, my dear friends, important only in glycolysis. BPG shunt, Rappaport-Lewis cycle, please try to understand, basically denotes oxygen unloading at peripheral tissue. Oxygen unloading at peripheral tissue. Important point to be noted. Shift to right. Oxygen hemoglobin curve shift to right. Shift to right. Oxygen hemoglobin curve shift to right. Fructolysis, liver cytosol, liver cytosol. Net gain fructolysis is always 7 ATP. Net gain is always 7 ATP. Fructolysis में net gain is always 7 ATP. Fructose is more dangerous than glucose, more atherogenic than glucose because of three things: hypercholesterolemia, hypertriglyceridemia, also more chance of gout, hyperuricemia. Essential fructosuria, fructokinase deficiency, aldolase B deficiency leads to HFI, hereditary fructose intolerance. Hereditary fructose intolerance. Sorbitol deposition, snowflake cataract in diabetes mellitus. Sorbitol deposition, snowflake cataract in diabetes mellitus. Galliput deficiency leads to galactosemia, also to some extent galactokinase. Galactose 1-phosphate uridyl transferase. Galactose 1-phosphate uridyl transferase. Important point to be noted. Oil drop cataract, galactosemia. Compound galactitol, dulcitol. Culprit enzyme due to aldose reductase. Due to aldose reductase. Due to aldose reductase. Aldose reductase is the culprit enzyme that is causing oil drop cataract. Oil drop cataract. Please remember classical features of galactosemia. Please try to remember, my dear friends. Day 3, day 4. Day 3, day 4. Important point. Day 3, day 4. Day 3, day 4. Please remember. Also, unconjugated hyperbilirubinemia. Child refuses to suck breast milk but accept glucose water. Breastfeed is contraindicated. Important question. Do not miss these lines. Galactosemia is important, my dear friends. Please try to understand. Right? Important point to be noted, my dear friends. Please remember, galactosemia is an expected question even in your upcoming exam. Do not miss this. Do not miss this. Important point to be noted. Human being can't produce vitamin C due to deficiency of L-gulonolactone oxidase. Gulonolactone oxidase. Coming to Garrod's tetrad, alkaptonuria, albinism, cystinuria, and essential pentosuria. Garrod's tetrad. Garrod's tetrad. Garrod's tetrad. Important point to be noted. Please remember, 4 fates of pyruvate. You already know: acetyl-CoA, lactic acid, then we have alanine, and also glucose by gluconeogenesis. These are the 4 fates of acetyl, 4 fates of pyruvate. 4 fates of pyruvate. Pyruvate to acetyl-CoA, oxidative decarboxylation. Pyruvate to acetyl-CoA, oxidative decarboxylation by PDH complex. PDH complex. Please try to understand. Produces 5 ATP. 5 ATP because of 2 NADH plus H plus. Link reaction. Multi-enzyme complex. PDH complex. Multi-enzyme complex. Please try to understand. There is no involvement of pyruvate decarboxylase. Which enzyme it does not consume? That is pyruvate decarboxylase enzyme is not included in this. Pyruvate decarboxylase. Pyruvate decarboxylase. Important. What are the important vitamins? B1, then we have B2, then B3 and B5, and non-vitamin coenzyme is lipoic acid. Lipoic acid. Lipoic acid. So please remember, pyruvate decarboxylase is not a part of this. B1, B2, B3, B5 and lipoic acid. Important point to be noted, my dear friends. Enzyme not related to, this is the line, remember, not related to PDH complex, pyruvate decarboxylase, right?
PDH complex deficiency, my dear friends, important point to be noted, high anion gap metabolic acidosis. And LP ratio, normally it is 6 to 10, but please try to understand in cases of PDH complex deficiency, this LP ratio will be increased. LP ratio. This ratio serves as a marker of tissue hypoxia in circulatory and septic shock. Important point to be noted. Try to remember this. Irreversible steps of Krebs cycle to citrate synthase alpha KGDH complex. Please try to understand, my dear friends. Per turn of Krebs cycle will produce 10 ATP, 10 ATP, 10 ATP, 10 ATP. Vitamins playing important role in Krebs cycle B1, B2, B3, B5. Vitamins, Krebs cycle B1, B2, B3, B5. Substrate level phosphorylation, we have succinate thiokinase. Complete aerobic oxidation of 1 glucose 32 ATP. Total number of isomer 32. Total number of ATP 32. Aerobic 7, PDH complex 5, citric acid cycle for 1 glucose Krebs cycle turns twice, so 20, total 32. Complete oxidation of 1 molecule of pyruvate 12.5 ATP. Inhibitors of Krebs cycle, FAM blockers, fluoroacetate, then we have arsenite, then we have malonate. FAM blockers, fluoroacetate, aconitis, arsenite, alpha KGDH complex, malonate, succinate dehydrogenase. Please try to understand. The successful heme, the mnemonic we have studied, my dear friends, the successful heme. What is successful heme? Important, anaplerosis, compensation of the lost intermediates. What is that? Please remember, succinyl-CoA, successful heme, succinyl-CoA can be generated by valine, isoleucine, methionine and threonine. The successful heme. Please try to understand, it can generate succinyl-CoA. My dear friends. Please remember, important point to be noted. Acetyl-CoA, ketone bodies, cholesterol and fats can never be converted to glucose. Never, my dear friends, can never be converted to glucose. Important point. Try to remember this, my dear friends. Amphibolic pathway, Krebs cycle. Amphibolic pathway. Please try to understand. Glycogen, density concentration wise, liver stores more glycogen. Liver, it is more concentrated. Please try to understand. End product of glycogenolysis in muscle is glucose 6-phosphate due to deficiency of glucose 6-phosphatase. Second time during the revision we are discussing. Please try to understand. Muscle glycogen is used in muscle only and it is basically maintaining muscle glycogen. It is mainly maintaining. Please try to understand the ATP's need. For ATP production. 8 ATP. How many ATP, my dear friends? 8 ATP in glycolysis. This glycolysis that is following muscle glycogenolysis, 1 ATP extra, 8 ATP. Whereas the liver glycogen, my dear friends, please try to understand, maintains normoglycemia up to 18th hour of fasting. Please try to understand, up to 18th hour of fasting. Glycogen, please try to understand, important point to be noted. The role of two different glycogen. Please remember, glycogen is the source of energy in 100-meter sprinter. That's why long distance marathon runners never ever compete with the sprinters. Marathon runners will never ever dare to compete with Usain Bolt. Usain Bolt, on the other hand, will never ever take participation in marathon running. Yes, because in case of sprinter, glycogen after the first minute, muscle glycogenolysis is important, right? Important point to be noted, my dear friends. Enzyme common to both glycogenesis and glycogenolysis, phosphoglucomutase. Phosphoglucomutase. Coming to glycogen storage disorder, important point to be noted, my dear friends. This chart you have to remember, we have type 0 Lewis hepatic glycogen synthesis. Most important of the lot, we are marking in green color, type 1a von Georg disease, glucose 6 phosphatase. Type 2a Pompey's acid maltase. Please try to understand. Type 3 and type 4 mnemonic ABCD for 3 and 4. ABCD for 3 and 4. Corey-Forbes disease, it is debranching enzyme. Anderson disease branching enzyme. Right. Type 5, my dear friends, important, most common in adult, McArdle's disease, muscle GP, muscle glycogen phosphorylase. Type 6, we have Hirsch disease, hepatic, H for H, hepatic glycogen phosphorylase. Tarwi's disease, muscle phosphofructokinase. Try to understand. Muscle phosphofructokinase, right? Important point to be noted. Type 7, Tarui muscle phosphofructokinase. Most common in children, my dear friends. Also, overall answer is von Georg's. Adults change the answer to McArdle's. Yes, children von Georg's. Adults McArdle's. Children von Georg's. Adults McArdle. Children von Georg's. Adults McArdle. Children von Georg's, adults, McArdle. Important point to be noted. Please try to understand. Von Georg disease we have seen fasting hypoglycemia, lactic acidosis, hyperlipidemia, ketosis and hyperuricemia. Yes, we have seen this, my dear friends. We have seen this, yes. Later on we have pot belly, doll-like faces, xanthoma formation. However, our liver enzymes ALT, AST are normal. Liver enzymes are never ever elevated in von Georg's disease. This is von Georg's disease. Coming to most fatal, my dear friends, Pompey's. Now treated by enzyme replacement therapy by L-glucosidase alpha. McArdle disease, important point. Exercise-induced myoglobinuria. Yes, cola-colored urine is passed by the patient, and there is also elevation of CK3, CKMM. Yes, important point to be noted. McArdle's disease. Muscle embodiment not seen. Muscle not 01A6. Abnormal type of glycogen, please try to understand, 3 and 4. Whereas cirrhosis, fourth one is more common than 3. And Cori cycle, C4C cytosol, and it is conversion of lactate. Muscle lactate is transported to the liver for gluconeogenesis. Cori cycle lactate, Cori cycle cytosol lactate, Cori cycle cytosol lactate, Cori cycle cytosol lactate, Cori cycle cytosol lactate. Yes, important point to be noted, my dear friends. Coming to glut. Rani Lakshmibai made India successful. Important question, let us quickly see, my dear friends. GLUT5 fructose absorption. GLUT4, my dear friends, insulin dependent. GLUT2 pancreatic beta cell non-insulin dependent. Rani Lakshmibai made India successful. Most abundant glut in these tissues as per 1, 2, 3, 4 and 5, 6. Yes, important point. Unidirectional transporters, my dear friends. Please remember, SGLT1 intestine absorption of glucose. Co-transporter of glucose in intestine is sodium. Co-transporter. SGLT2 kidney tubules. defect gene mutation of SGLT2, renal glycosuria. Renal glycosuria, renal glycosuria. SGLT1, intestine. Please remember, sodium is the co-transporter. Both of them secondary active transport. Secondary active transport. Important point to be noted. SGLT1 secondary active transport. SGLT2 kidney tubules, renal reabsorption defect, renal glycosuria, renal glycosuria, renal glycosuria, renal glycosuria, renal glycosuria, SGLT2. SGLT2. Important point to be noted. Try to understand, my dear friends. HMP shunt maximum in liver, maximum in liver, maximum in liver, my dear friends. Rate limiting enzyme G6PD, 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 glucose 6-phosphate dehydrogenase. Yes. Transcatolase dependent on B1, transcatolase. Important point to be noted. Maximum NADPH concentration is in lens. Maximum NADPH concentration is in lens. Important point to be noted. Functions of NADPH, most important function, fatty acid synthesis, then glutathione reduced one, cholesterol bile acids, and also respiratory burst. Important point to be noted, my dear friends. Now, please try to understand. Important point to be noted. NADPH prevents hemolysis. So, G6PD deficiency that generates NADPH. Please try to understand. If reduced glutathione is less, please try to understand, we can have hemolytic anemia. Most common cause of hemolytic anemia in India is G6PD deficiency followed by pyruvate kinase deficiency. Important point to be noted. Detoxification of H2O2 within the RBC was done by selenium dependent glutathione peroxidase. Assessment of B1 deficiency is done by measuring RBC transcatolase activity, where B2 deficiency RBC glutathione reductase activity. B1 RBC transcatolase, B2 RBC glutathione reductase. Yes. What is glutathione? What is glutathione? What is glutathione? What is glutathione? GCG, tripeptide. GCG, glutamate cysteine glycine, and it is a pseudopeptide. Gamma glutamyl cystinyl glycine. Helps which enzyme? GGT, gamma glutamyl transferase, gamma glutamyl transferase. Important point to be noted. Sulfa group of drugs are to be avoided in G6PD deficiency. Please try to understand. Differentiation between G6PD deficiency and pyruvate kinase deficiency. Heinz bodies in peripheral blood smear. Heinz bodies due to condensation of hemoglobin. Wernicke-Korsakoff syndrome, my dear friends, we have talked about. Wernicke-Korsakoff syndrome, B1 deficiency. Isolated encephalopathy, Goa, never forget Goa. Global confusion, ophthalmoplegia, ataxia. Please try to understand. Goa. Important point to be noted. Goa, important. Psychosis when confabulation present together in a patient. Please remember, most common of B1 deficiency in India is alcohol, daru, khamba. Please remember, most common cause of B1 deficiency in India is chronic alcoholism. If you have alcohol as the cause of B1 deficiency, you have syndrome together. But if you have other cause of B1 deficiency like eating polished rice, we have isolated Goa. Psychosis alone not present. Please remember, all these features with sympathetic overactivity, answer changes to delirium tremens. tremens. Important point to be noted, my dear friends, right? Coming to quickly a understanding of the rate limiting enzymes. Rate limiting enzyme, although there is a very less chance that they will be asking you questions on rate limiting enzymes, still if anyone is interested to remember all these things in one shot, I have summarized most 15 most important pathways whose rate limiting enzymes are mentioned here. At least I have to see the most important ones. Glycolysis, PFK1, gluconeogenesis, 1,6 bisphosphatase, fructose 1,6 bisphosphatase, right? Krebs cycle, alpha ketoglutarate dehydrogenase complex, better answer than isocitrate dehydrogenase. Glycogenesis, glycogen synthase, genolysis, glycogen phosphorylase. HMP shunt, G6PD, 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 glucose 6 phosphate dehydrogenase. Cholesterol synthesis, HMG CoA reductase, HMG CoA reductase. Ketone body synthesis, HMG CoA synthase, HMG CoA synthase. Fatty acid synthesis, acetyl CoA carboxylase, acetyl CoA carboxylase. Beta oxidation, CAT1, 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 beta oxidation. Urea cycle, CPS1, CPS1, urea cycle. Tyrosine metabolism, tyrosine hydroxylase. Bile acid synthesis, 7 alpha hydroxylase, and purine synthesis, PRPP glutamyl amidotransferase. At least we have discussed all these first 13 in our discussion of biochemistry, whether face to face or online classes. We have discussed, my dear friends. You may get one question from this, but usually the chance of this is less. Okay, moving further. Insulin increases glycogenesis and decreases gluconeogenesis. Question, recently asked question. Insulin decreases gluconeogenesis. Important point to be noted. Decreases gluconeogenesis. Coming to amino acids and proteins. Yes, important point to be noted, my dear friends. Please try to understand, right? Amino acid, most important points. Let us try to, we all know these points that we have discussed. This is the basics that we already know. I'm not marking any highlighter point from here. Try to focus these ones. Branched chain amino acids, VIL, valine, isoleucine, leucine. Sulfur containing, methionine, cysteine. Two cysteine will combine to form cysteine. Try to understand. Hydroxyl group containing amino acid, tri-3-sari, tyrosine, threonine, serine. Tri-3-sari. Basic amino acids. His basics are loose. Histidine, arginine, lysine. Lysine is, please try to understand, arginine here is the most basic. Lysine loose. His basics are loose. Basics for basic amino acid, histidine, arginine, lysine. Arginine most basic. Aromatic amino acid, HTTP, web browser, histidine, tyrosine, tryptophan, phenylalanine. Maximum UV absorbance by tryptophan. Please remember, uncharged polar, good souls are truly the God's children. Glycine, serine, R is not arginine. This is asparagine. Tyrosine, threonine, glutamine, and cysteine. Yes, good souls. Purely 100% ketogenic, it is leucine. Lay tai fish, mixed ones. Lay tai fish, lysine, tyrosine, tryptophan, phenylalanine, and isoleucine. Lay tai fish. Essential amino acid, TV till 8 p.m. Essential amino acid, TV till 8 p.m. Please try to understand, my dear friends. TV till 8 p.m. Important point to be noted. Please remember, do not miss this, my dear friends. TV till 8 p.m. TV till 8 p.m. Please try to understand. Please remember, histidine contains imidazole ring, blood buffer. Hemoglobin also act as a buffer due to presence of histidine. Histidine. Important point to be noted. Albuminuria, heat and acetic acid test. Heat and acetic acid test. Albuminuria, heat and acetic acid test. Important point. Important point. We have discussed. Please try to understand, my dear friends. Biuret technique for serum total protein estimation. Biuret technique for serum total protein estimation. Important point to be noted, my dear friends. Biuret technique for serum total protein estimation, my dear friends. Please remember, overall strongest bond is peptide bond. Strongest peptide, weakest van der Waals force. Van der Waals force. Please try to understand. Hydrogen bonding guides into folding process. Guides into protein folding process. Most common amino acid alpha helix, alpha ala. Please try to understand. And best source of beta plated sheet is silk fibroin. Proline breaks both alpha helix and beta plated sheet because it is causing kinking or bending. Kinking or bending. Important point to be noted, my dear friends. Tertiary structure, multiple weak interaction. Most important of all of them is van der Waals forces. Weakest of all of them is, or contributing minimum hydrophobic interaction. Although van der Waals is weakest but contributes maximum, hydrophobic bond is contributing minimum towards stability of the tertiary structure. Right, important point. Single polypeptide chain always end with tertiary structure. Recently asked question, albumin, myoglobin. Myoglobin, recently asked question, my dear friends. Please remember, skin keratin is softer than nail keratin due to less number of disulfide linkage. Less number of disulfide linkage. Please try to understand. Prion, proteinaceous infective particle. Normal primary but abnormal tertiary due to improper or abnormal beta plated sheet. Important question. Secondary structure that is abnormal in case of prion protein is beta plated sheet abnormal. Most common human form, it is Creutzfeldt-Jakob. Cerebral cortex become like sponge. Alzheimer's and Parkinson's both are due to abnormal folding but not prion disease. What are the abnormal proteins? Abnormally folded proteins in Alzheimer's are hyperphosphorylated tau, and we have studied also beta amyloid precursor protein. Right, important point to be noted. Most abundant protein in our body is collagen. Please try to understand. Most abundant plasma protein, albumin, and glycine is the most abundant. Glycine is the most abundant amino acid found in collagen. Most abundant amino acid found in collagen, my dear friends. Please try to understand. Collagen, three individual chains together, triple helical structure tropocollagen. Procollagen to collagen conversion is done by hydrogen bonding between hydroxyproline and hydroxylysine. Please try to understand this. Vitamin C takes part in post-translational modification in collagen formation. So scurvy, please try to understand scurvy, defective hydroxylation, important point to be noted, gum bleeding, teeth loss, petechial hemorrhages, collagen cross linking, lysyl oxidase, dependent on copper. Important point. Recently asked question, Menkes kinky disease, ATP7A gene mutation, copper transporter malfunction in the GI tract. Important lines are important. Finally, defective aldol condensation, and we will be having kinky or curly hair. Important point to be noted. Kinky or curly hair, my dear friends. Kinky or curly hair. Types of collagen. Remember mnemonic scab. Type 1, skin, bone, also joint capsule. Type 2, cartilage, vitreous humor of eye. Type 3, we have vascular tissues, arteries, skin also. Type 4, basement membrane. Scab. Skin, type 1, type 2, cartilage, type 3, artery, type 4, basement membrane. Scab, SCAB, scab. Collagen vascular disorders, important point, my dear friends. Type 1, osteogenesis imperfecta. Please try to understand. Frequent fractures in bone, brittle bone disease, and blue sclera. Important point. Important point. Type 2, chondrodysplasia. Type 3, Ehlers-Danlos syndrome. Most common defect, type 5, but in most severe there is defect of type 3. Hyperstretchable skin and hyperextensible joints. Frequent bruising with minor trauma. They can pull their skin like this. Hyperstretchability in the skin. Type 4 defect, Alport syndrome. Most common, hematuria. Also, glomerular basement membrane, basket whip appearance, and sensory neural hearing loss. Type 6 and 7 defect together, epidermolysis bullosa, frequent blisters formation. Bullosa blisters. Try to understand. Apart from this, we have also Marfan syndrome. You remember the mnemonic Marfan. Kunal sir has taught you. Marfan syndrome, mnemonic Marfan. Also collagen vascular disorder. Right. Please try to understand. Protein folding assisted by chaperones. Please try to understand, my dear friends. Heat shock proteins. We have seen HSP 70, HSP 60. 60 is also known as chaperonin. If you can stretch, you can remember few others. Protein disulfide isomerase or peptide prolyl cisterase isomerase. Otherwise, remember only HSP 70 and HSP 60. Please try to understand. Right, important point to be noted, my dear friends. Please remember this. Coming to metabolism of amino acids. Important point to be noted, my dear friends. Please try to understand. Kwashiorkor, edema, fat sugar, baby. Also, please try to remember we have voracious appetite. Also seen in case of kwashiorkor. Voracious appetite. It is seen in case of kwashiorkor only. Voracious appetite. Although marasmus, they are devoid of carbohydrate, protein and fat, but actually patients of marasmus, they suffer from anorexia. The drive to eat food is lost because they have been still surviving without carbohydrate, protein and fat. So the urge to eat food is gone. Voracious feeders are kwashiorkor children. Important point to be noted. Although kwashiorkor is isolated protein deficiency, but we have voracious appetite in them. Important point to be noted. Try to understand. Marasmus, monkey face, skin on bone appearance. Skin on bone appearance. Sometime also asked a question, flaky paint appearance of skin. Answer is again kwashiorkor. Flaky paint, flaky, F-L-A-K-Y, flaky paint type of appearance, kwashiorkor. Important point to be noted. Please remember, question has come on your exam. Mr. Cycle, glutathione dependent, absorption of neutral amino acid from intestine. Absorption of neutral amino acids from intestine. Important point to be noted. Please remember this. Ubiquitin mark of death. Ubiquitin mark of death. Important point to be noted. Hyperammonemia can lead to sequestration of alpha ketoglutarate because transamination dependent on alpha ketoglutarate. So the chance of Krebs cycle may be damaged. So please try to understand. Krebs cycle in neurons can be damaged in patients of hyperammonemia. Hyperammonemia. It will take out alpha ketoglutarate from the Krebs cycle so that it can be used in transamination reaction. Try to understand, my dear friends. Transamination reaction. Important point to be noted. Please remember. In aerobic condition, next level of amino acids that are seen at the level of pyruvate are alanine and glycine. Alanine and pyruvate, they are converted by transamination. Important point to be noted. Increased level of serum alanine after fasting means there is mobilization of the muscle proteins followed by increased gluconeogenesis. We have seen this. Increased gluconeogenesis, my dear friends. Please try to understand, my dear friends. Brain and other tissue detoxify ammonia as glutamine, whereas muscle utilizes alanine and participates ultimately in glucose-alanine cycle or Cahill cycle. Cahill cycle is gluconeogenesis of alanine. Cori's cycle is gluconeogenesis of lactate. Right, important point to be noted, my dear friends. Urea cycle PUBG we have already seen. Please remember, CPS1, the rate limiting enzyme, please try to understand. Allosteric activator is NAG, N-acetyl glutamate. N-acetyl glutamate. And carbamyl phosphate serves as fuel for urea cycle, sustains urea cycle, right? Important point to be noted. Ammonia is the donor of the first amino group in urea and aspartate is the donor of the second amino group in urea. Ammonia and aspartate are the sources of nitrogen atom in urea biosynthesis, right? Important point to be noted, my dear friends. Please remember, arginine is degraded to urea by arginase, and during the same step it also generates ornithine. Yes, latest question we have seen breakdown of arginine, but during that synthesis of ornithine produces urea. If you break arginine, you will get urea, but at the same time ornithine is released. Important point to be noted, my dear friends. Please try to understand. 3 ATPs are required for 1 urea synthesis. 3 ATPs. Urea bicycle, fumarate goes out, aspartate comes in. Fumarate is a better answer. Coming to urea cycle disorders, my dear friend. Urea cycle disorders. Please remember, urea cycle disorder. Type 1 CPS1 deficiency, not so much important. Type 2 OTC deficiency, yes. Moderate hyperammonemia, we have glutamine increased in blood, orotic aciduria, bouncer question we have discussed. Glutamine in blood, orotic aciduria, answer type 2. Type 3 transporter defect, ornithine transporter defect, triple H syndrome. triple H syndrome, hyperornithinemia, hyperammonemia, and homocitrullinuria. Homocitrullinuria. Yes, 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 yes. Please remember. Now, specialized products obtained from tyrosine, tyrosine give rise to, please remember the mnemonic ATM, ATM, ATM. A stands for mainly adrenaline, adrenaline. Along with that, also remember other catecholamines. We have dopamine and noradrenaline or norepinephrine. Also, please remember T stands for T3, T4 and M stands for melanin. ATM, remember ATM, 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 tyrosine ATM, adrenaline, catecholamines series, thyroid hormones, melanin. Also those glucogenic and ketogenic compounds like fumarate and acetoacetate, but that is not so much important. Remember the mnemonic ATM, 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 tyrosine, ATM, tyrosine, ATM, tyrosine, ATM, tyrosine, ATM, tyrosine. Yes, important point to be noted. Tyrosine hydroxylase. We have seen rate limiting enzyme of catecholamine synthesis as well as overall or entire tyrosine metabolism. Overall tyrosine hydroxylase. Yes. Yes, 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 yes. Catecholamine synthesis, C synthesis is dependent more on C than B. Yes. Yes, we know this. Catecholamine synthesis, it is mainly dependent on C. C for C, I told you, C for C. Catecholamine synthesis is dependent more on vitamin C than B6, Please remember. Norepinephrine to epinephrine, noradrenaline to adrenaline is just by simple addition of methyl group by Sam uncle. Methyl group by Sam uncle. If you can stretch, please remember the full name of this methyl transferase, that is phenylethanol-amine N-methyl transferase. At least remember methyl transferase, addition of methyl group with norad so that it can be converted to adrenaline. Yes. Please remember. Phenylketonuria, my dear friends, classical, non-classical, classical phenylalanine hydroxylase deficiency, non-classical dihydrobiopterin reductase deficiency. Important point. Most common inborn error in metabolism overall. Most common inborn error overall. Please try to understand. Please remember, classical PKU, severe mental retardation, idiot, IQ of an idiot, IQ of an idiot, IQ of an idiot, PKU, classical type, medical emergency, yes. Phenylketones like phenyl acetate, mousy, musty odor in PKU, mousy, musty odor in PKU. Mousy, musty odor in PKU. Please remember, blondes are seen in PKU. Blondes are seen in PKU. Blondes are seen in PKU. Best technique to detect IEM, tandem mass spectroscopy. Tandem mass spectroscopy. Tandem mass spectroscopy. PKU, if phenylalanine level in the plasma or serum is more than 20 milligram per deciliter. Best treatment modality in PKU is to stop phenylalanine rather than adding supplementation with tyrosine. So tapioca can be given, biopterin can also be added. Yes. Type 1 tyrosinemia, fumarate acetoacetate hydrolase deficiency, cabbage or mushroom like body odor. Cabbage or mushroom like body odor. Important point to be noted. PHPP dioxygenase deficiency, we have neonatal tyrosinemia. PHPP dioxygenase neonatal tyrosinemia, vitamin C supplementation, vitamin C supplementation, vitamin C supplementation. Swimming pool odor, Hawkinsinuria, swimming pool odor, Hawkinsinuria, swimming pool odor, Hawkinsinuria. Important point to be noted, my dear friends. Homogentisate oxidase deficiency leads to alkaptonuria. Homogentisate oxidase leads to deficiency, leads to alkaptonuria. Benzoquinone acetate gets deposited, ultimately deposited as polymerized form as alkaptone. Uchronosis, blackening of the soft tissues. Pinna of ear, pinna of ear. Also remember underneath the palm, palmoplantar pigmentation, image based questions, palmoplantar pigmentation, zone of calcification in X-ray, lumbosacral spine, zone of calcification in X-ray, lumbosacral spine. Alkaptonuria, frequently asked question. Please try to understand. We also have cola colored urine during transition and black color urine on full color change. So urine color changes on standing in alkaptonuria. Alkaptonuria. Flag type of hair, not vitamin, copper deficiency. Flag type of hair. Tyrosinase albinism. Tyrosinase albinism, right? Flag type of hair, copper deficiency. Specialized products obtained from tryptophan, serotonin, melatonin, vitamin B3, niacin. Serotonin, melatonin, vitamin B3, niacin. Serotonin, melatonin, vitamin B3, niacin. Niacin sparing, it will spare. Tryptophan will spare vitamin B3. Even if you don't take, it will produce it in the liver. 60 milligram produce 1 milligram. 60 milligram of tryptophan produces 1 milligram. 60 is to 1. 3% goes for B3 production. B6 deficiency can also lead to pellagra. B6 deficiency can lead to pellagra. Kynureninase was dependent on B6 or PLP. Please try to understand. Xanthurenic aciduria seen in B6 deficiency. And the best diagnostic modality to measure B6 deficiency will be RBC transaminase activity. RBC transaminase activity. Melatonin production is dependent on SAM. Melatonin SAM uncle dependent. Argentophenoma or malignant carcinoids, they are most commonly originating from ileum rectum appendix. Presents with intermittent diarrhea, flushing and sweating as well as pellagra like symptoms. Diagnosis, estimation of 5 here. Estimation of hydroxyindole acetic acid in the 24-hour urinary output collection. Yes, important point. Coming to Hartnupp's disease. Hartnupp's disease. Please remember, defective transporter for tryptophan, mostly for tryptophan. Hartnup disease mainly for tryptophan, mostly for tryptophan. Important point to be noted. 4Ds of pellagra, you know. Dermatitis, classically, Cazal's necklace. Important point, please remember. Methionin's active form, Sam uncle. Universal methyl group donor. Yes, universal methyl group donor. We know Mr. C3P3 RND or MELT C3P3 RND. Mr. C3P3 RND, melatonin, creatine, carnitine, catecholamine, 3P, protein, phosphatidylcholine, polyamine. Then we have RNA, DNA and neurotransmitter. Yes, one more time, Mr. C3P3 RND, we have seen melatonin, creatine, carnitine, catecholamine. Then we have protein, phosphatidylcholine, that is also known as lecithin, polyamine, RND, RNA, DNA, center, neurotransmitter. Yes, important point. This we have talked about. Mr. C3P3 RND. MELT also you can remember so that you can remember melatonin. Right, important point. Homocystinuria is due to deficiency of cystathionine beta synthase, classical type, and also due to deficiency of B6, B9, B12. Also due to deficiency of B6, B9, B12. B6, B9, B12. Please remember. Folate trap. B12 deficiency causes functional folate deficiency. B12 deficiency causes functional folate deficiency. Folic acid is getting trapped as methyl tetrahydrofolate because to take out that methyl group, we require vitamin B12 to come and take out that in a bi-bi reaction manner. If B12 is missing, B9 will get added with methyl group. but it cannot give it to homocysteine. Homocysteine to methionine conversion, it is not done by SAM, it is done by vitamin B9 and B12 together. If B12 is missing, even if you have B9 in our body, but it will be trapped as methyl tetrahydrofolate, right? Important point. Homocystinuria, very, very important. They look as Marfan, but also show other great complications. Yes, they look as Marfan, but also show other great complication. Tall stature, lens dislocation, accelerated atherosclerosis, Marfanoid habitus and mental retardation, blue eyes, arachnodactyly, common feature with Marfan syndrome as well, seizures, stroke, also common with Marfan syndrome, osteoporosis, common feature with osteogenesis imperfecta, blue eyes also, please try to understand, complication common with osteogenesis imperfecta, G say glaucoma, C say cataract, and Charlie Chaplin gate. They look as Marfan but also show other great complications, right? Important point to be noted. Please try to understand. The most direct precursor of taurine is cysteine. Cysteine से बनता है taurine and glutathione. Right? Glutathione also. Cola urine, cystinuria. Cola cystinia. Cola colored urine, alkaptonuria, McArdle's disease, myoglobinuria, or post streptococcal glomerulonephritis. But cola urine, answer changes to cystinuria. My dear friends, please try to understand. Histidine forms histamine by non-oxidative decarboxylation. It is also a vasodilator along with mast cell stabilizer, right? Figlu excretion test, also known as histidine loading test, folic acid B9 deficiency assessment. Yes. BCAA, branched chain amino acid metabolism, valine isoleucine leucine, not seen in liver, mainly in skeletal muscle. Maple syrup urine disease, BCKD. Maple syrup, MSUD BCKD. Branched chain ketoacid dehydrogenase complex. Yes, important. Burnt sugar smell in urine, DNPH test. Burnt sugar smell in urine, DNPH test. Dinitrophenyl hydrazine test. Important point to be noted, my dear friends. Please remember, isovaleric acidemia, sweaty feet odor. Isovaleric acidemia, sweaty feet odor. Glycine, most important coin. A heart full of careless sentiments usually fails badly. Yes. We know the meaning of this mnemonic. Yes. Heart full of careless sentiments usually fails badly. Please remember, H, heme synthesis, F, flexibility of proteins, O, optically inactive, careless, creatine and collagen synthesis, sentiments, smallest, simplest. Usually we have 3U, purine, glutathione, hippuric acid synthesis. FA, fails, we have folic acids required for its own synthesis. BA, bile acid conjugation. Bile acid conjugation also requires taurine. So please remember, heart full of careless sentiments usually fails badly. What is creatine? It is a tripeptide GAM, glycine, arginine, methionine, or SAM. Important point to be noted, my dear friends. Glycine, arginine, methionine, or SAM. Important point to be noted, my dear friends. Please remember, important point. Please remember. Creatine phosphate, energy store in the muscle, Lohmann's reaction. Yes, Lohmann's reaction. Creatine phosphate, energy store in the muscle, Lohmann's reaction up to first minute. Yes, beyond first minute, muscle glycogenolysis. Up to first minute, creatine phosphate. Beyond first minute, muscle glycogenolysis. arginine, nitric oxide, NOS, needs for speed. NOS, nitric oxide synthase, NOS. Please remember, important point to be noted. 3 NOS. Most important NOS is NOS3, endothelial NOS, EDRF. Please remember, vasodilator, endothelium derived relaxing factor, NOS3, most common one. EDRF inhibits also platelet aggregation and under the control of calcium. NOS1, my dear friends, it is in brain, neuronal NOS, brain, controlled by calcium, neurotransmitter. NOS2, my dear friends, it is in macrophages. Bactericidal killing of bacteria, and it is under the control of not calcium by interleukin 1 and TNF alpha. Yes, cytokines, interleukin 1 and TNF alpha. Remember, recently asked question, nitric oxide activates the enzyme guanylyl or guanylate cyclase, and the second messenger that is released in response to nitric oxide is cyclic GMP, and this causes dephosphorylation, not phosphorylation. Phosphorylation is done by cyclic CAMP camp. This is not camp. CGMP, that is dephosphorylation of myosin light chains and causes vasodilation. So second messenger CGMP, guanylate cyclase, enzyme responsible dephosphorylation, ultimately causing myosin light chains to get separated from each other and vasodilation. Yes, important question that has been asked recently in your exam. Nitroglycerin or glycerol trinitrate tablet, it is given in sublingual manner for treatment of pain in angina pectoris, because it increases nitric oxide's availability in the buccal mucosa. Yes. Polyamines are three: putrescine, spermidine and spermine, but not cadaverine. Polyamines are already derived from SAM. Mr. C3P3 RND, 1P was polyamine. Fish odor syndrome, trimethylaminuria. Flavin containing monooxygenase deficiency. Fish odor syndrome, trimethylaminuria. Coming to lipids. Lipids, TAG, stored ones, are broken down by hormone sensitive lipase in a G protein coupled receptor manner. Hormone sensitive lipase. After the release from the adipose tissue, please remember, they are carried in the plasma by albumin as Z protein. Please try to understand. Carnitine shuttle, we have seen, it is applicable only for long chain fatty acids. Only for long chain fatty acids. What is carnitine? It is not fat. It is a protein derived mainly from lysine and methionine in the liver, and this synthesis of this is also dependent on vitamin C. So carnitine dependent on vitamin C. Catecholamine dependent on vitamin C. C for C. Collagen dependent on vitamin C. C for C everywhere. Please try to understand, my dear friends. Right, carnitine. Important point. Lysine, methionine. That is also from Sam uncle. CAT1 location, I told you, of the enzymes of carnitine shuttle. Location, CAT1 outer membrane, but translocase and CAT2 are inner membrane. Yes, CAT1 location wise questions are asked. CAT1 outer membrane, but translocase and CAT2 are inner membrane. Please try to understand, my dear friends. Inner membrane. Please remember. Coming to this complete beta oxidation of 1 palmitic acid. Complete beta oxidation of palmitic acid. Please try to understand. Complete beta oxidation of one molecule of palmitic acid produces 106 ATP. Palmitic acid 106. Remember the formula 7N minus 6. And stearic acid, that is an 18 carbon, produces 120 ATP. 120 ATP. Important point to be noted, my dear friends. MCAD deficiency, medium chain acyl CoA dehydrogenase. Medium chain acyl CoA dehydrogenase. Medium chain acyl CoA dehydrogenase. Just a moment, my dear friend. Just a moment. Yes. Please try to understand this. Medium chain acyl CoA dehydrogenase, MCAD. This is the most common inborn error of lipid metabolism, but second most common overall. And the deficiency of this causes some pathetic disorder, sudden infant death syndrome, SIDS. Please try to understand. SIDS, SIDS. What is SCID? SCID is severe combined immunodeficiency due to ADA deficiency. This is SIDS, sudden infant death syndrome due to medium chain acyl CoA dehydrogenase deficiency. Most common from lipids. Second most common overall, phenylketonuria, right? Yes, coming to this. Jamaican vomiting sickness, hypoglycine A. Jamaican vomiting sickness, hypoglycine A. Zellweger syndrome, cerebrohepatorenal syndrome, paroxysmal beta oxidation defect. Paroxysmal beta oxidation defect. Zellweger syndrome, paroxysmal beta oxidation defect. Cerebro, progressive damage of brain, liver, kidney. First is brain, then liver, then kidney. Yes, important point to be noted. Zellweger syndrome. Please remember, excess PUFA accumulation in brain causes hypomyelination. Repsum disease, problem with paroxysmal alpha oxidation. Phytanic acid storage disease. Master blaster question. M for master blaster. Methyl malonic aciduria is seen only in B12 deficiency. Differentiating feature between B9 and B12. Methyl malonic aciduria only with B12 deficiency, not with B9 deficiency. So B1 deficiency, RBC transcatolase. B2 deficiency, RBC glutathione reductase. B6 deficiency, those xanthurenic aciduria or RBC transaminase. B1 RBC transcatolase. B2 RBC glutathione reductase. B6 RBC transaminase. B9 Figlu excretion test or histidine loading test. B12 methyl malonic aciduria, where we have methyl malonyl CoA coming out in urine. Right? Important point to be noted, my dear friends. Please try to understand. Fatty acid synthesis take place in cytosol, whereas beta oxidation was in mitochondrial outer membrane, then through inner membrane, carnitine shuttle, then in the matrix. Yes. Please try to understand. Well-fed state, fatty acid synthesis is inhibited because well-fed state fatty synthesis is promoted, while beta oxidation will be inhibited as malonyl CoA inhibits CAT1. Which one is inhibited? CAT1. When? Well-fed state. By whom? Malonyl CoA. What is malonyl CoA? Intermediate of fatty acid synthesis. Yes. So during well-fed state, we have to talk about fatty acid synthesis. During starvation, we have to talk about beta oxidation. So in a well-fed state, malonyl CoA inhibits CAT1 so that beta oxidation is blocked so that fatty acid synthesis can take place. Yes. Essential fatty acid. It's essential fatty acid. Please try to understand. Essential fatty acid. Linoleic, alpha linolenic. Please try to understand. Then we have arachidonic, icosapentaenoic, and docosahexaenoic. These five. Linoleic is the most essential. Alpha linolenic is the prototype of omega 3. Prototype of omega 3. Please try to understand. Linoleic safflower oil, source safflower oil. Alpha linolenic flaxseed linseed oil. Arachidonic acid, meat. EPA in fish oil or fish fat, and DHA in breast milk. Right? Arachidonic acid contains 4 double bonds. EPA 5, DHA 6. Arachidonic acid contains 4 double bonds. Ideal ratio of omega 6 is to omega 3 is 4 is to 1. But to achieve this, we have to add more of omega 3 because our principal food is rice, which is already giving us huge amount of omega 6 than what we require. Right, in flour oil. Please try to understand. Maximum permissible limit for trans fat intake is less than 1% out of total fat intake per day. Trans fat is the worst type of fat that any human being can consume. But we Indians are proud of consuming that in a heavy proportion. Yes. Unfortunate. Please try to understand. Eicosanoids are derived from 20 carbon arachidonic acids. Icosa 20 carbon. Icosa 20 carbon. Although EPA, eicosapentaenoic acid, also 20 carbon, but these eicosanoids, prostaglandin, prostacyclin, thromboxane, and leukotrienes, they are derivative of arachidonic acid. Arachidonic acid. Please try to understand. Glycolipids does not contain phosphate. Anticardiolipin antibodies are actually seen in TB, malaria, syphilis, and APLA syndrome. Syphilis, recently asked question. Please do not forget. Syphilis, anticardiolipin antibodies. Lecithin, most abundant phospholipid of our plasma membrane. And if with this lecithin, if you add 2 palmitic acid, dipalmitoyl phosphatidylcholine or dipalmitoyl lecithin, please remember these are actually working as surfactant produced by type 2 pneumocytes. LS ratio more than 2 indicates mature lung. Checked usually between 28 to 32 weeks of gestation. Checked in between 28 to 32 weeks of gestation. Please try to understand. Platelet activating factor, apart from activating platelets or causing platelet degranulation and activation, it is also important in acute inflammatory reaction, hypersensitivity, and anaphylactic reaction. Important point to be noted. Both plasmalogen and platelet activating factor are ether linked compound. Cephalin, that is phosphatidyl ethanolamine, needed in blood clotting. Cephalin blood clotting. Phosphatidylserine, marker of apoptosis. Right, important point. Gangliosides like GM1 acts as cell surface receptor for entry of cholera toxin or tetanus toxin in our intestinal mucosa. 2020 FMG exam question in the month of August. Yes. GM1, entry of cholera toxin and tetanus toxin. Important point to be noted, my dear friends. Phospholipase A2, snake venom, viper snakes, hemolytic agent because it produces lysolecithin. Only difference between blood group A and B is N-acetyl group, that is present in only in group A. N-acetyl group only in group A. Rancidity means basically deterioration of fats that can be prevented by vitamin E. Rancidity prevented by vitamin E. That is the deterioration of fats, right? Lysosomal storage disorder. Most common Gaucher's disease, beta glucosidase, Niemann-Pick's sphingomyelin, Krabbe 2B, so this is beta galactosidase, Fabry, only one B, alpha galactosidase A, metachromatic leukodystrophy, aryl sulfatase A, Farber's disease, ceramidase, Tay-Sachs, hexosaminidase A, Sandops, both hexosaminidase A and B. Yes, one more time. Gaucher's, beta glucosidase, Niemann-Pick, sphingomyelin, Krabbe's disease, beta galactosidase, Fabry's disease, alpha galactosidase A, metachromatic leukodystrophy, aryl sulfatase A, Farber's disease, ceramidase, then Tay-Sachs disease, hexosaminidase A, and we have Sandops disease, hexosaminidase A plus B. Yes, you can also remember. Yes, just try one more time. Gaucher's, beta glucosidase, Niemann-Pick, sphingomyelin, Krabbe's, beta galactosidase, Fabry, alpha galactosidase A, metachromatic leukodystrophy, aryl sulfatase A, Farber's, ceramidase, Tay-Sachs, hexosaminidase A plus B, answer changes to Sandops disease. Yes, you guys can remember. I have immense belief in all of you, right? Cherry red spot in macula, cherry tree never grow tall in sands. C is crow, CRAO, cherry red spot in macula. Cherry tree never grow tall in sands, CRAO. Ketone bodies are ABA. What is ABA? ABA, acetoacetate, beta hydroxybutyrate, and acetone. Right. Important point. Ketone bodies are produced from acetyl CoA during mainly starvation, and maximum organ or maximum utilization of ketone bodies is done by brain. Liver although synthesizes but cannot utilize this due to deficiency of thiophorase. Thiophorase is not seen in liver, seen in tissues like brain, muscle, heart, kidney. That's why it will produce in liver ketone bodies, all the ABA will produce there, but it will utilize here because of presence of this here, not in the liver. Yes, important point to be noted. Ketosis, ketonemia, ketonuria, and smell of acetone breath. We can do something known as ketonuria detection by Rothera's test or dipstick test. Rothera's test. It is done bedside test, 2 rupees test. Ketonuria detection. Yes, right. Please try to understand. In starvation, there is hypoglycemia, lipolysis, lipid breakdown, beta oxidation, ketoacidosis due to production of ketone bodies, but never ever hypercholesterolemia. Recently asked question. See this question that has come recently. Starvation में hypoglycemia, lipolysis, lipid breakdown, ketone body synthesis, ketoacidosis is seen, but not hypercholesterolemia. Hypercholesterolemia, cholesterol synthesis is seen in a well-fed state, not in starvation. Yes, not in starvation. Cholesterol is never elevated in case of starvation. It is hypoglycemia, lipolysis, ketone body overproduction, but not except important point. Please remember this. Cholesterol, 27 carbons are derived from acetyl CoA. LCAT. Who is LCAT? We have seen. It is present in the blood, and what does it do? Free cholesterol to cholesterol ester in the plasma. Free cholesterol to cholesterol ester in the plasma under the guidance of HDL. Third carbon of cholesterol was containing one hydroxyl group that will be getting esterified by getting a fatty acid from lecithin. Yes, important point. Cholesterol synthesis cytosol. C for C. Cholesterol cytosol. Cholesterol synthesis cytosol. Enzyme common to both cholesterol and ketone body synthesis? Answer is HMG-CoA synthase. Answer is HMG-CoA synthase. HMG-CoA synthase. Important point to be noted, my dear friends. Cholesterol helps in the production of, my dear friends, bile acids, bile salts, vitamin D, and steroid hormones like ACTH, but not the bile pigments, except bile pigments. Except bile pigments. Bile pigments are not coming from cholesterol. Those are not coming from cholesterol, my dear friends. Please try to understand. Bile acids and bile salts are coming from cholesterol, but not the bile pigments. Yes, not the bile pigments, right, beta? Important. Lipoprotein chemistry. Lipoprotein chemistry. Please remember, chylomicrons, largest in size, maximum TAG. Please try to understand. Carry exogenous TAG from small intestine to the peripheral tissue. VLDL, endogenous fat from liver to the peripheral tissue. LDL, maximum concentration of the cholesterol, atherosclerosis and coronary artery disease. HDL, good cholesterol, reverse cholesterol transport and cardioprotective. Best marker of dyslipidemia, LDL HDL ratio more than 2 precipitates atherosclerosis. LDL is beta, IDL broad beta, VLDL is pre-beta and HDL is alpha lipoprotein. Segregated by Frederickson's method of electrophoresis. LDL beta, HDL alpha, IDL broad beta, VLDL pre-beta. VLDL pre-beta, yes. HDL, please try to understand, there is no ApoB, deficit in ApoB. HDL, remaining all apoproteins are present except ApoB, neither ApoB 100 nor ApoB 48. B48 is present in the chylomicron. Chylomicron. B100, it is present in LDL, IDL, VLDL. Only apoprotein present in LDL is B100. Whereas IDL also contains ApoE. So we have seen uptake of IDL in the liver for LDL synthesis happens via ApoE. ApoE. That is the question we have marked. Right. Please try to understand the ApoB48 and ApoB100, they are actually derived from a single gene. But during the process of RNA editing, they will give rise to different functional biological product. They are coming from same gene, but ultimately they will give rise to two different type of proteins due to a process called RNA editing. RNA editing. ApoE, arginine-rich protein. 3 is the most common type, but 4th is MCQ, Alzheimer's disease. ApoE4 is associated with Alzheimer's disease. ApoE4 associated with Alzheimer's disease. Lipoprotein small a, little rascal, chota sa, nanna munna sa, rascal, LPA, little rascal. Please remember, significant structural similarity with plasminogen blocks fibrinolysis process in vivo and highly atherogenic. We Indians unfortunately are having higher levels of lipoprotein small a. ApoA1 is an activator of LCAT and ApoC2 is activator of lipoprotein lipase. Yes, A1 activates LCAT, A2 inhibits LCAT, ApoC2 activates lipoprotein lipase or LPL. Lipoprotein lipase, it is absent in liver and maximum in heart. Maximum in heart. Adult liver does not have lipoprotein lipase. Where it is present? Towards the peripheral tissue, not towards the liver. So, highest concentration is in heart. Right. Important point to be noted. So, during starvation, usually the fats are mobilized from adipose tissue to the heart for beta oxidation. Type 1 hyperlipoproteinemia, chylomicron syndrome. TAG very high while cholesterol normal. due to defect in lipoprotein lipase or ApoC2 deficiency. But my dear friends, important is the second one. Type 2A, very, very important, marking in green color. Very, very important, type 2A. This is primary familial hypercholesterolemia. Very important. Primary familial hypercholesterolemia. This is the most common, and this is the defect of LDL receptor. TAG normal, but LDL will be huge, huge. It can even cross 1000 milligram per deciliter, where normally the target is less than 100 milligram per deciliter. Incompatible with life, most life-threatening, autosomal dominant, tendon xanthoma, corneal arcus, xanthelasma. This is very, very important. Type 2A, expected question. Type 2A, expected question. Please remember this. Familial LCAT deficiency, fish eye disease. Familial LCAT deficiency, fish eye disease. Important point to be noted. Tangier's disease, HDL close to nil. HDS close to nil. We have orange yellow tonsils. Orange yellow tonsils. We have seen orange yellow tonsils. Orange yellow tonsils. Tangier's disease. Nucleotide chemistry, please try to understand, only focusing on the just important points, my dear friends. We have uric acid as the end product of purine degradation. Uric acid as the end product of purine degradation. Important point to be noted. Gout, monosodium urate crystals. Monosodium urate crystals, needle shaped ones in our synovial cavity that we have to extract by arthrosynthesis. By arthrosynthesis. Right. Most common joint involved during acute attack of gout. It is our first metatarsophalangeal joint or big toe known as podagra. Podagra. Indian word podagra. Most common joint involvement during acute attack of gout. First metatarsophalangeal joint. Right. Podagra. Important point to be noted. Leishman syndrome, purine salvage pathway defect, HGPRT, hypoxanthine guanine phosphoribosyl transferase. Child has unopposed urge to destroy themselves, self-mutilation, self-mutilations. They can present with orange sands in diaper, orange sands in diaper, but orange crystals in diaper, answer will change to orotic aciduria. Orotic aciduria. Pyrimidine metabolism defect, OPRT, orotate phosphoribosyl transferase and OMP decarboxylase. Leishman syndrome is more important in your upcoming exam. Do not miss this. Important point to be noted. Right. Vitamins we have already touched with the major pathways. However, just focusing on few important points from vitamins. If you can stretch, please remember. B1 deficiency, you know, very. Wet beriberi, wet. We call it wet beriberi because of edema. Edema is seen in wet beriberi. We have high output cardiac failure. High output cardiac failure. But in dry beriberi, there is no edema. Only peripheral neuritis. In dry beriberi, yes. B2 deficiency, my dear friends, we have angular stomatitis, glossitis, cheilosis. We have also circumcorneal vascularization, and we have basically vascularization where a lot of blood vessels are formed around the cornea, circumcorneal around the cornea, and they can present also with seborrheic dermatitis. Niacin B3 causes pellagra. Please try to understand. Maize and sorghum eaters, they can lead to have high chance of pellagra. B6 deficiency can also cause pellagra. Drugs like isoniazid, heart nerve disease, argentophenoma or malignant carcinoid syndrome, please try to understand. They can also cause pellagra. Please remember, B3 has been used to reduce the level of the little rascal. We have just now seen little rascal. Little rascal is this lipoprotein small a that has been seen can be decreased in us if we take B3 supplementation more. Important point to be noted. B5 deficiency, burning feet syndrome. B6 deficiency, we have got sideroblastic anemia. Also try to understand, as B6 can be stored in our body, 80% can be stored as a part of glycogen phosphorylase. It is the only water soluble vitamin with significant toxicity, and the significant toxicity is basically sensory neuropathy. It is basically sensory neuropathy. Please remember, B6 has a toxic status. Toxicity of B6 causes sensory neuropathy. B6, sensory neuropathy. Sensory neuropathy. Please try to understand. Coming to B7, we know avidin that is present in raw egg decreases biotin. Please try to understand. Avidin that is found in raw egg is decreasing biotin absorption. That's why we should not never ever consume raw egg. Don't crack and drink egg. No, don't do that. Cook it semi-solid or boil it or omelette, right? Important point. Avidin present in raw egg decreases biotin absorption, and biotin deficiency can cause multiple carboxylase deficiency because biotin is important for carboxylation process. Most common vitamin deficiency in India is B9 deficiency, that is basically seen with having macrocytic anemia. You know this. Please remember, B12 deficiency is seen in strict vegan diet. All of you know this. B12 is also known as extrinsic factor of Castle, that will bind with intrinsic factor further. So B12 extrinsic, B12 है extrinsic factor of Castle, right? Important point to be noted. B12 and folic acid deficiency both leads to macrocytic anemia, whereas B12 deficiency can lead to megaloblastic anemia and pernicious anemia, and also development of both upper motor neuron and lower motor neuron lesion like features. That's why we have another disorder called subacute combined degenerations, SCD, not SCID. SCID is severe combined immunodeficiency. I is missing. This is SCD, subacute combined degeneration, only with B12 deficiency. Only with B12 deficiency, right? Important point to be noted. Methyl malonic aciduria we have seen only with B12 deficiency. Please try to understand. B12 deficiency, we have also pernicious anemia, where we have autoantibodies generated against intrinsic factor in the parietal cells and parietal cells, right? Assessment of B12 deficiency can also be done by a test known as Schilling test, where we use radioactive leveled cobalt 60. Cobalt 60 is a question, my dear friends. Schilling test, cobalt 60. Assessment of B12 deficiency. Please try to understand. We have also seen vitamin C was first brought into the picture because of disorder of scurvy, first disorder in the whole world, and Sir James Lind proposed treatise on scurvy. Vitamin C helps in the iron absorption. We have seen this question, by keeping iron in ferrous state for absorption, and reduces also folate to form tetrahydrofolate, that is the active form, right? Vitamin C supplementation is also important for wound healing, ulcer treatment, in trauma, burns treatment, as an antioxidant. A, C and E, S complex, vitamin A, vitamin C, vitamin E are antioxidant, right? Important point to be noted. Also decrease the incidences of common cold, cataract and carcinoma bladder. Vitamin C. Vitamin A deficiency, please try to understand, it can lead to night blindness, xerophthalmia, and in severe cases this xerophthalmia can be further degraded to form corneal melting, that is known as keratomalacia. And also the triangular white patch deposition over the cornea is called as Bitot spots, right? Important point to be noted, right? Biologically important vitamin A is 11-cis retinal, that you would have also heard during rhodopsin cycle, or also known as world's visual cycle. What is rhodopsin? 11-cis retinal plus opsin. Rhodopsin is 11-cis retinal plus obscene. Important point to be noted, my dear friends. Please try to understand. And the main key excitatory response in our visual pathway or world visual cycle is not depolarization. This is hyperpolarization is the key excitatory response to the visual cortex. Not depolarization, but hyperpolarization. Yes. Please try to understand. Active form of vitamin D is calcitriol. Calcitriol 125 dihydroxycholecalciferol, which is derived from the rate limiting enzyme that is 25 alpha hydroxylase in the liver. Yes. Followed by final activation in the kidney by 1 alpha hydroxylase. So, 2 times hydroxylation after the dehydrocholesterol was formed. Please try to understand. First is 25 hydroxylation in the liver, that is the rate limiting step, and we have further 1 alpha hydroxylation that is in the kidney, final activation, calcitriol. Please try to understand, my dear friends, if you can stretch and remember, vitamin D deficiency also is detrimental for coronary artery disease because it increases something known as CACS, coronary artery calcium scoring, that is known as Agatston scoring. If you can stretch only, then remember this, otherwise leave it, right? Vitamin E, most potent antioxidant, anti-fertility vitamin, and also prevents rancidity. We have discussed during our revision class as well, right? Quench lipid peroxidation. Quench or prevents lipid peroxidation. Vitamin E deficiency can cause hemolytic anemia in the newborn, as well as resorption of fetus and testicular atrophy. Vitamin E deficiency can cause hemolytic anemia of the newborn. Hemolytic anemia of the newborn. Resorption of fetus and testicular atrophy. That's why anti-infertility vitamin. This deficiency will cause testicular atrophy. Important point. Whereas vitamin K deficiency is the only fat-soluble vitamin with a coenzyme function needed for post-translational modification of factor 2, 7, 9 and 10. Water-soluble, please try to understand, synthetic form is menadione. Water-soluble form, menadione. Yes, we have discussed this. And please remember, vitamin K deficiency can cause hemorrhagic disorder of the newborn. Vitamin E deficiency, hemolytic anemia of the newborn. This one is hemorrhagic disease of the newborn, right? And in vitamin K deficiency, prothrombin time will be prolonged, right? Coming to minerals, please try to understand. Although we have discussed about the cofactor topic, please remember, molybdenum, we know, xanthine oxidase, maximum it is found in the liver. Please remember, zinc is needed for synthesis, storage, stabilization and secretion of insulin, where chromium potentiates action of insulin. Important question. All other things about insulin mark the answer as zinc. So remember this, potentiates. All S, synthesis, storage, stabilization, secretion, answer is zinc for insulin. But potentiation of insulin action, answer changes to chromium. Yes, important point, my dear friends. A child with alopecia, hyperpigmentation and hypogonadism and dermatitis of both oral cavity as well as genital region, please remember, is suffering from zinc deficiency, and this sort of dermatitis and the disorder of zinc deficiency is known as acrodermatitis enteropathica. Acrodermatitis enteropathica. Please try to understand. Important point to be noted. Now, selenium deficiency leads to Khashan's disease. Please try to understand. Selenium deficiency, Khashan's disease. Please try to understand this. And please try to understand, selenium and vitamin E, they go hand in hand. They are best friends of each other. So selenium will decrease the need of vitamin E, and vitamin E will further need or decrease the need of selenium, and vice versa. Please try to understand. Important point to be noted, my dear friends. Please try to remember this. Miscellaneous topics we have already seen various times. Just remember that succinyl CoA plus glycine will produce delta ALA. Rate limiting enzyme of heme synthesis is ALA synthase. ALA synthase. B6 PLP dependent. B6 PLP dependent. If possible, if you can remember, if you can stretch, only then follow the following points, my dear friends. Please remember, we have lead poisoning basically inactivates ferrochelatase enzyme and it can prevent heme synthesis. Most common porphyria in India is Porphyria cutanea tarda. But if they ask you a question, most common acute porphyria, answer will change to acute intermittent porphyria. Acute intermittent porphyria. And please remember, my dear friends, in acute intermittent porphyria, please try to understand, my dear friends, important point. These patients, they will present with acute abdominal pain and neuropsychiatric symptoms. Yes, acute intermittent porphyria or AIP, acute intermittent porphyria or AIP, the patient will present with acute abdominal pain and neuropsychiatric symptoms. These two are important points, recently asked question in your exam. At least try to remember this. Porphyria, that is the disorder on the heme biosynthetic pathway. First enzyme was ALA synthase. This deficiency causes sideroblastic anemia or B6 deficiency. But this one, most common acute porphyria is acute intermittent porphyria. Here there is a deficiency of the enzyme PBG deaminase, porphobilinogen deaminase or hydroxymethylbilane synthase, and it presents with abdominal pain and neuropsychiatric symptoms, right? Please try to understand, my dear friends, right? Not going into the detail of those points of the molecular biology. Molecular biology we have not touched, so I am not going to talk about this. If any student is interested to go through this, they can go by themselves, but I am not going to focus on the questions of molecular biology. So with this, we have actually done the quick revisions of this, my dear friends. Quick revision. In just 2 hours time, we have actually been able to revise the entire biochemistry. We have quickly gone through. Let me help you, my dear friends, one more time to go through this content in a quick span of time this time, so that we will just quickly brush through the most important points. Just try to understand most important points. Right. Now I am marking them with another green color highlighter. Let me just help you on going through this important point. Just try to understand most important points one more time. Already highlighted yellow ones are important. Furthermore important. Yes, let us quickly revise one more time. Quickly. Multiple rapid revisions are required for you to remember the questions. At least solve more than 80 to 90% of the questions that will be coming from biochemistry from this content. Let us focus on denaturation. Primary structure stable, 2, 3, 4 lost. 2, 3, 4 lost. Important point. Focus on this one, green color, changing the color now. Most important ones we are focusing. Blotting technique, snowdrop. Blotting technique, snowdrop. Southern DNA, northern RNA, western protein, eastern enzymes or post-translational modification of proteins. Please try to understand. Important point to be noted. Please try to focus. Oxidative decarboxylation by B1. B6 non-oxidative decarboxylation. Please try to understand carboxylation B7, gamma carboxylation vitamin K, most important ones. One carbon pool, vitamin B9, one carbon pool, do not miss. And acyl group carrier coenzyme A, acyl group carrier B5 coenzyme A. Right. Factor 2, 7, 9, 10, vitamin K dependent. zinc containing enzyme SOD carbonic anhydrase ALP ADA. Remove dehydrogenase now. SOD carbonic anhydrase ALP ADA. Quickly let's focus on the most important points. Clinical importance of the ALP, my dear friends. Clinical importance of the ALP. Please try to understand. Drastically high bone disease. Obstructive jaundice is moderately or very high levels, we call it sometime up to 10 to 12 times. Moderate, we have hepatic disease. Hepatic disease. Isolated GGT, my dear friends. Isolated GGT, we have alcoholic liver disease. ADA deficiency, we have SCID. First successful gene therapy. First successful gene therapy. Try to understand. Celluloplasmin peroxidase in the plasma. Wilson disease, deficiency of celluloplasmin. ATP7B gene mutation. Most important we are targeting now. Most important. Those who are looking for very limited content, this time is for you. Again, one more time, we are doing the quick revision with the most important points. Yes. Hepatolenticular degeneration. KF ring, sunflower cataract, chalcosis. Chalcosis. Right. Important point to be noted. Mitochondrial SOD manganese. Yes, important point. Please remember them. Selenium containing enzyme, do not miss them. Glutathione peroxidase, thyroidine reductase, deiodinase. Selenocysteine, 21st amino acid derived from serine coded by UGA. Important question. Please remember. Peripheral conversion of T4 to T3, D2 deiodinase. Xanthine oxidase, molybdenum containing. Km, substrate concentration at Vmax by 2. Taq polymerase, PCR, magnesium, important point, magnesium. RT-PCR, amplification of RNA. Now I am targeting those MCQs which has been asked in very recent times. Yes. One more time we are doing the quick revision. RT-PCR, amplification of mRNA. Important point to be noted. Please try to understand. Allosteric, Km increase, Vmax decreased. Vmax decrease. Try to understand, my dear friends. Isoenzymes catalyze same reaction. LDH1 heart flipped pattern. Important point to be noted. LDH3 lungs, COVID-19 patients. LDH4 most abundant in pancreas, may be asked this time. And LDH5 and 4, both if they ask you, most abundant in liver and skeletal muscle, answer is both 5 more than 4. Yes, important point. Try to remember this, my dear friends. Creatine kinase, CKMB, marker of choice for reinfarction. Right. Lactate dehydrogenase. Please try to understand. It is a marker for cytoplasm. Lactate dehydrogenase, marker of cytoplasm. Biological oxidation, important point. Please remember, up to first minute, creatine phosphate. After first minute, muscle glycogenolysis. Important point to be noted. Beta subunit produces ATP. Beta subunit of F1 of ATP synthase complex 5 produces ATP. Right, important point to be noted. Please remember, blockers. How can we forget Alka? Right, Alka cheers rotten pears in bar. Don't miss this. Do not miss this. Carboxyl malonate, these two, and we have got these four. These four. Do not miss them. Oligomycin complex 5 inhibitor. Oligomycin complex 5 inhibitor, my dear friends. Please try to understand. Cyanide blocks cytochrome A3, non-competitive inhibition. Cyanide blocks cytochrome A3, non-competitive inhibition. Uncoupler 2,4-DNP. Uncoupler 2,4-DNP. 2,4-DNP. Please try to understand. Thermogenin UCP1, most important physiological uncoupler. Most important physiological uncoupler. Atkin diet, LCHPK diet, low carbohydrate, high protein, ketogenic diet. Low carbohydrate, high protein, ketogenic diet. Please try to understand this, my dear friends. HbA1c more than 66.5%, diagnosis of diabetes. Advanced glycation end products. Advanced glycation end products. Advanced glycation end products. Important point to be noted, my dear friends. Hyaluronic acid, synovial fluid, vitreous humor of eye. Yes, dermatan sulfate, hard valves. Keratan sulfate, corneal transparency. Corneal transparency. Heparan sulfate, basement membrane. Chondroitin sulfate, aorta cartilage, most abundant gag. Most abundant gag. Right. Hurler syndrome, corneal clouding. Hunter syndrome, X-linked recessive. X-linked recessive. Right. Important point to be noted, my dear friends. Right. Right. Important point. Please remember. Please remember this. POCT, beta form of glucose only. POCT, beta form of glucose only. POCT, beta form of glucose only. Lactose intolerance, diarrhea, vomiting, abdominal cramp, child cries immediately after birth. Child cries immediately after birth, my dear friends. Child cries immediately after birth. Carbohydrate metabolism. End product of glycolysis in RBC, it is lactate. End product of glycolysis in RBC is lactate, right? Cancer cells derive nutrition from anaerobic glycolysis. Important point to be noted, my dear friends. Important point. Glucose 6 phosphate connecting link of entire carbohydrate metabolism. Carbohydrate metabolism. Important point. Please try to understand. Incorporation of inorganic phosphate, glyceraldehyde, 3-phosphate dehydrogenase. 3 irreversible steps. Do not miss. Do not miss these 3 irreversible steps, my dear friends. Please try to understand. Insulin promotes glycolysis by 3 key enzymes of glycolysis except hexokinase. Except hexokinase. Only reversible kinase, phosphoglycerate kinase. Phosphoglycerate kinase. Additive used for sugar estimation, it is sodium fluoride potassium oxalate. Sodium fluoride potassium oxalate, right? Important point to be noted, my dear friends. Please remember difference between HK, focus on hypothalamus. Recently asked question. Diabetes mellitus, HK4. Hyperglycemic states, HK4. Hypoglycemic states, HK2. Brain cells entry of glucose during starvation, HK2. HK2. Important point. Hemolytic anemia due to pyruvate kinase deficiency. Failure of sodium potassium ATPase pump. Failure of sodium potassium ATPase pump, right? Important point to be noted. Do not forget PUBG. Do not forget PUBG, right? Liver is the most common site for gluconeogenesis. Parle-G, do not forget Parle-G. Alanine contributes maximum. Alanine contributes maximum, my dear friends. Alanine contributes maximum. Yes. Propionate, glucogenic fatty acid, main one. Right. Important point to be noted. Glucose 6 phosphatase absent in muscle, no gluconeogenesis in muscle. No gluconeogenesis in muscle. Right. Important point to be noted, my dear friends. Please try to understand this. Right. Right. Mallet shuttle only in glycolysis. Only in glycolysis. BPG shunt, oxygen unloading at peripheral tissue. Shift to right. Shift to right. Shift to right. Right. Important point to be noted. Fructolysis always 7 ATP. Fructolysis always 7 ATP, my dear friends. Please try to understand this. Right. Fructose more atherogenic than glucose. Why? These three. Please try to understand. Essential fructosuria, fructokinase deficiency, aldolase B, we have got hereditary fructose intolerance. Most common age of presentation, we know it is after 5 to 6 months. After 5 to 6 months. Sorbitol deposition, snowflake cataract in डायबिटीज मेलाइटस. गैलेक्टोसीमिया भूल के भी मिस मत करना. गैलेक्टोसीमिया, गैलीपुट मोर देन गैलेक्टोकाइनेस. This oil drop cataract culprit enzyme name is aldose reductase. Aldose reductase. Please try to understand. Presentation on day 3, day 4. Unconjugated hyperbilirubinemia, refusal to suck breast milk but accepting plain glucose water. Breastfeeding contraindicated. Do not miss galactosemia. Important point to be noted, my dear friends. Important point to be noted. PDH complex, multi-enzyme complex, B1, B2, B3, B5, lipoic acid. Pyruvate decarboxylase is not a part. We have seen this question already. Not related to PDH complex. Please remember pyruvate decarboxylase. Pyruvate decarboxylase, right? PDH complex deficiency, HAGMA, LP ratio increased, NADH NAD plus ratio decreased, NADH NAD plus ratio decreased. Normally it is 6 to 10, but in PDH complex deficiency this will be increased LP ratio, right? Important point, marker of tissue hypoxia. Irreversible reactions of the Krebs cycle, citrate synthase, alpha ketoglutarate dehydrogenase complex. Please try to understand. Per turn, we have got 10 ATP. 10 ATP. Vitamins B1, B2, B3, B5. Vitamins B1, B2, B3, B5. Substrate level phosphorylation, succinate thiokinase. Total production 32. Total production for aerobic oxidation by glucose, we have got 32. 1 pyruvate 12.5, 12.5, 12.5. Please remember the mnemonic called successful Vim. Threonine, valine, isoleucine, methionine can compensate for the loss of succinyl CoA if it goes out for heme synthesis. Yes, important point to be noted. Please remember, acetyl CoA, ketone bodies, cholesterol, fats never get converted to glucose. Amphibolic pathway. Answer is Krebs cycle. Krebs cycle is amphibolic pathway. Which enzyme of the Krebs cycle is not seen in the mitochondrial matrix? Succinate dehydrogenase. Not given in this all-in-one section, but please remember that as well if you can. Yes, try to understand, my dear friends. Glycogenolysis in the muscle is glucose 6-phosphate due to deficiency of glucose 6-phosphatase in the muscle. Muscle glycolysis, that is seen during starvation following glycogenolysis, it generates, my dear friends, 8 ATP. 8 ATP. And liver glycogen is able to meet the need of those glucose maintenance. It can do the glucose maintenance normoglycemia, my dear friends, up to 18th hour of fasting. Up to 18th hour of fasting. Yes. Yes, important point. Common enzyme to glycogenesis, glycogenolysis, phosphoglucomutase. Phosphoglucomutase. Von Geerk's disease, do not miss. Von Geerk disease, already highlighted in green color. Do not miss. Do not miss, my dear friends. Do not miss. Most common Von Geerk's in children overall. McArdle's adults. Please try to understand. Do not miss the clinical feature of von Geerk's disease. Do not miss normal ALT AST. Important point. McArdle's disease, exercise-induced myoglobinuria. Also, there is increase in the level of CK3 or CKMM. Important point to be noted, my dear friends. Cori cycle is in cytosol, lactate, Cori cycle, lactate. Right, important point to be noted, my dear friends. Please remember, fructose absorption, GLUT5, insulin dependency on GLUT4, insulin independent GLUT2. Please try to understand. Secondary active transport, both SGLT1 and SGLT2. Co-transporter is sodium, do not miss. Renal glycosuria, do not miss. SGLT2 gene mutation. SGLT2 gene mutation. Important point. Transcatolase is dependent on B1 TPP. Maximum NADPH concentration is seen in lens. Maximum. Right, beta? Important point to be noted. Please try to understand this NADPH, my dear friends. Reduced glutathione production will be increased and H2O2 detoxification by selenium dependent glutathione peroxidase. Most common cause G6PD deficiency followed by pyruvate kinase deficiency. RBC, transcatolase, B1, B2, RBC, glutathione reductase. Do not miss this, right? Glutathione, GCG, glutamate, cysteine, glycines. Important point. Please remember this, my dear friends. Please remember, Heinz bodies only G6PD deficiency anemia. Heinz bodies only G6PD deficiency anemia. Wernicke-Korsakoff syndrome, B1 deficiency, either only Goa or after going to Goa, confabulation as well. If the cause of B1 deficiency is alcohol, do not miss Wernicke-Korsakoff syndrome. Chronic alcoholism as well as narcotic abuse. Right, important point. Rate limiting enzyme, as I told you, most likely they are not going to get us, so I'm not marking. Important point. Please remember, decreased gluconeogenesis by insulin. Insulin decreases gluconeogenesis, but mainly it is anabolic hormone. Glycogenesis, lipid synthesis, and protein synthesis. Amino acid, yes. Important. At least remember VIL, branched chain. Then focus on tri-3-sari. His basics are loose. HTTP. Good souls are truly the God's children. 100% ketogenic leucine. 100% ketogenic leucine. Also try to remember, my dear friends, we have got late tai fish, both glucogenic and ketogenic. TV till 8 p.m. Essential amino acids. Hemoglobin can act as buffer. Hemoglobin can act as buffer. Hemoglobin can act as buffer at physiological pH due to presence of histidine. Heat and acetic acid test, albuminuria, detects albuminuria, right? Important point to be noted, my dear friends. Please try to understand. Proline destabilizes both alpha helix and beta pleated sheet by introducing kinks or bends. Important point, my dear friends. Please remember this. Hope there is no confusion here, right, beta? Please remember this, yes. Tertiary structure stabilized by multiple weak interaction. But out of those multiple weak interaction, most important is van der Waals forces. Single polypeptide chain myoglobin. Recently asked question. Skin keratin is softer due to less number of disulfide linkage. My dear friends, please try to understand. Prions, proteinaceous infective particle, they have improper beta pleated sheet. Most common human form, Creutzfeldt-Jakob disease. Alzheimer's and Parkinson's, they are not prion disease. They are due to abnormal folding, not prion disease. Hyperphosphorylated tau. Question. Hyperphosphorylated tau. Alzheimer's. Also remember beta amyloid precursor protein. Here. Important point to be noted. Please try to understand. Glycine is the most abundant amino acid found in collagen. Most abundant amino acid found in collagen. 3 alpha chains. Please try to understand. Every third amino acid is glycine. Every third amino acid is glycine. Please try to understand. Pro-collagen to collagen conversion. Vitamin C is needed for post-translational modification. Vitamin C is needed for post-translational modifications. Scurvy, gum bleeding. Collagen cross-linking, lysyl oxidase, dependence on copper, ATP7A gene mutation, Menkes kinky disease, copper transporter is malfunctioning in the GI tract, defective aldol condensation, curly kinky hair. Recently asked questions, do not miss that topic. Try to gather info for that particular topic. Do not miss Menkes kinky hair disease. Coming to types of collagen, my dear friends, please try to understand this. Types of collagen: scab. Recently asked question: skin, joint capsule, vitreous humor, basement membrane. Recently asked question. Please remember them, right? Type 1, osteogenesis imperfecta, blue sclera, brittle bone disease. Type 3, Ehlers-Danlos syndrome. Most common type is type 5, but in most severe type 3. Remember alphabet E. E is the fifth alphabet. Right, important point. Hyperstretchable skin. Hyperstretchable skin, my dear friends. Hyperstretchable skin. Important point to be noted. Right, important point. Please try to understand. Chaperones assisting protein folding process, heat shock proteins, HSP70, HSP60. 60 is also known as chaperonin. Please try to understand, my dear friends. Please try to understand. This kwashiorkor marasmus, multiple time getting repeated. One more time, kwashiorkor, edema due to isolated protein deficiency, fat sugar baby, they are fed only with carbohydrate. So excess carbohydrate will be deposited as fat sugar baby, and they will be having voracious appetite. Seen more with kwashiorkor. Marasmus children may suffer from anorexia as well. Yes, type of question can be asked as a bouncer. Please try to understand, my dear friends. Monkey face, skin on bone appearance is for marasmus. Voracious appetite, kwashiorkor. Yes, important point. Please remember. Mr. Cycle has been recently asked. Mr. Cycle for absorption of neutral or non-polar amino acids in intestine. Do not miss this. Do not miss this. Hyperammonemia can inhibit Krebs cycle because of sequestration of alpha ketoglutarate and going towards transamination. Yes, important point to be noted, my dear friends. Please remember, very, very important. Increased level of serum alanine after fasting. This suggests mobilization of the muscle proteins and increased gluconeogenesis. Brain and other tissues detoxify ammonia as glutamine, and muscle detoxify is at alanine. Do not miss this point. Cahill cycle is for alanine, also known as glucose-alanine cycle. Yes. CPS1 allosteric activator is N-acetyl glutamate. Right, important point. Ammonia and aspartate are the source of nitrogen atoms for urea synthesis. Ammonia and aspartate are the source of nitrogen atom for urea synthesis. Try to understand. Arginine breakdown by arginase, while generating ornithine produces urea. Latest question. Do not miss this, my dear friends. 3 ATPs are required for 1 molecule of urea, and urea cycle and Krebs cycle are linked by mainly fumarate. Urea bicycle. Yes. Please try to understand type 2, my dear friends. Type 2, my dear friends. Type 2. OTC, glutamine in blood, orotic aciduria, bouncer type of question. Transporter defect for ornithine, triple H syndrome. Please remember the mnemonic ATM, ATM for the specialized products that can be obtained from tyrosine. Specialized products that can be obtained from tyrosine. A say adrenaline, that is mainly our catecholamine, also his other best friends, dopamine, noradrenaline, T3, T4, and M stands for melanin. Rate limiting enzyme of this pathway of catecholamine synthesis is tyrosine hydroxylase. More dependent on vitamin C. Catecholamine synthesis, more dependent on vitamin C. Please try to understand. Noradrenaline to adrenaline conversion is done by phenylethanol N-methyl transferase. So, methyl transferase. Methyl group is coming from Sam uncle. Sam uncle. Please try to understand. Phenylalanine hydroxylase. Phenylketonuria, classical type. Dihydrobaptene reductase, non-classical. Most likely they will ask you question from classical type. They are having idiots. IQ of an idiot. Classical PKU. Please try to understand. Mousy, musty odor in PKU due to phenyl acetate. Blondes are seen. No need to go to the parlor. The child will be having a defect of melanin synthesis. So, brown color hair, right? Blondes are classically seen. Tandem mass spectroscopy, gold standard technique for IEM detection. Inborn error in metabolism, my dear friends. Best treatment modality in PKU is to stop intake of phenylalanine. Tapioca can be given, which is having less phenylalanine but more of tyrosine. Stopping is more important than addition of tyrosine, my dear friends. Please try to understand. Type 1 tyrosinemia, cabbage or mushroom like body odor. Fumaryl acetoacetate hydrolase. PHPP dioxygenase neonatal tyrosinemia, vitamin C supplementation. Right, important point to be noted. Alkaptonuria, never ever miss. Homogenticate oxidase. Yes. Benzoquinone acetate is the actual compound that is polymerized to form alkaptone that is getting deposited in our soft tissues. Right. Important point. And causes something, my dear friends, known as black discoloration of the soft tissues, that is ochronosis. Most common site, pinna of ear. Then we have intervertebral cartilages, as well as knee joint, my dear friends, as well as cornea sclera, and underneath the palm, palmoplantar pigmentation. Yes, image based question. So, cola colored urine can be seen in patients of alkaptonuria. Ultimately, black color on full color change, but the patient passes normal color urine. Yeah. Flag type of hair, copper deficiency. Specialized products from tryptophan, serotonin, melatonin, and niacin. Please try to understand. MSN Explorer, MSN Explorer, melatonin, serotonin, niacin. Yes, important point to be noted. Niacin sparing amino acid is tryptophan. 60 milligram will give you 1 milligram of niacin. B6 deficiency can also cause pellagra. Please try to understand xanthurenic aciduria, B6 deficiency, and RBC transaminase activity. Melatonin production dependent on SAM uncle. Argentophenoma, ileum rectum appendix, intermittent diarrhea, flushing, sweating. Diagnosis by estimation of 5 HIAA. It can also present with pellagra like symptom. Argentophenoma also do not miss. Heart nerve disease, mostly for tryptophan. Dermatitis, Cazal's necklace in case of this. Image based question. Methionine's active form, SAM, universal methyl group donor. SAM, methyl group donor, universal methyl group donor. Remember, Mr. C3P3RND. One more final time, C3P3RND. Mr. M, melatonin. C, creatine, carnitine, catecholamine. 3P, protein, phosphatidylcholine, polyamine. Then we have RNA, DNA, neurotransmitters. Yes, great. Important point. Please remember them. Right, beta. Please remember them. Yes. Please remember. Yes. Homocystinuria, cystathionin beta synthase. Cystathionin beta synthase also can be done by deficiency of B6, B9, B12. Right. Folate trap, B12 deficiency, functional folate deficiency. Homocystinuria, do not miss, they look as Marfan but also show other great complication. Expected question. Homocystinuria is so common in India. Right. Please try to understand. Cola urine, cystinuria. Cola urine, cystinuria, my dear friends. Histidine to histamine by decarboxylation process. Figlu excretion test, folic acid deficiency. You may mark this one. BCAA metabolism, maple syrup urine disease, BCKD, burnt sugar, DNPH. Burnt sugar, DNPH, right. Important point to be noted, my dear friends. Glycine, do not miss, heart full of careless sentiments usually fails badly. Creatine is a tripeptide of CAM, glycine, arginine, methionine. Glycine, arginine, methionine. Arginine to nitric oxide by nitric oxide synthase. Please try to understand. Right, important point to be noted. Nitric oxide activates guanylate cyclase. Second messenger is cyclic GMP that causes dephosphorylation, my dear friends. Dephosphorylation of myosin light chains. Dephosphorylation of myosin light chains, that is causing vasodilation, right? Nitroglycerin or trilglycerine, glycerol trinitrate tablets, pain in angina pectoris by releasing more of nitric oxide, by activating NOS3. It activates NOS3, right? Polyamine, cadaverine is not a polyamine, right? Fish odor syndrome, trimethylaminuria. Trimethylaminuria. Please try to understand, my dear friends. Carnitine is derived from lysine and methionine. Lysine and methionine. CAT1, outer membrane. CAT2 and translocase, inner membrane of mitochondria. Right. Important point to be noted. Most common inborn error of lipid metabolism is sudden infant death syndrome, SIDS, sudden infant death syndrome. Jamaican vomiting sickness, hypoglycemia A, Zellweger syndrome, cerebrohepatorenal defect of peroxisomal beta oxidation. Please try to understand this, right? Important point to be noted, my dear friends. Please remember this. Coming to this one. Peroxisomal alpha oxidation, refsum disease, phytanic acid storage disease. Bouncer, master blaster question. Methyl malonic aciduria, B12 deficiency. Do not miss this. Do not miss this, my dear friends. Right. Important point to be noted. Malonyl-CoA inhibits CAT1 in a well-fed state. Malonyl-CoA will ensure fatty acid synthesis in a well-fed state by inhibiting beta oxidation, by blocking the rate limiting enzyme of beta oxidation, that is CAT1. That is CAT1, my dear friends. Important point to be noted, right? Please remember. Important point. Arachidonic acid, 4 double bonds, and eicosanoids are derived from this 20 carbon arachidonic acid. Ideal ratio 4:1. Trans fat less than 1%, maximum permissible limit. Right, important point. Anticardiolipin antibody or antiphospholipid antibody, syphilis. Lecithin can produce dipalmitoyl phosphatidylcholine, that is acting as a surfactant produced by type 2 pneumocytes. LS ratio more than 2 indicates mature lung. Important point. Cephalin, blood clotting. Phosphatidylserine, marker of apoptosis. GM1, cell surface receptor for cholera toxin and tetanus toxin. Important question that has been asked. Phospholipase A2, viper snake venom, hemolytic agent. Please try to understand, my dear friends. Lysosomal storage disease, you have to remember. We never know which one will be asked. So please try to understand. Try to remember this one more time, my dear friends. Gaucher's disease, beta glucosides. Sphingomyelinase deficiency, Niemann-Pick disease. Beta galactosidase, Krabbe's disease. Alpha galactosidase A, Fabry's disease. Aryl sulfatase A, metachromatic leukodystrophy. Farber's disease, ceramidase deficiency. Tay-Sachs and Sandoff's. Tay-Sachs is hexosaminidase A and Sandoff's is hexosaminidase A plus B both together. A plus B both together, my dear friends. Please try to understand this. Yes, hope you're able to follow this, right? Cherry tree never grow tall in sands. Do not miss. Do not miss this. Please remember. Ketone bodies are broken down during starvation mainly in brain. Mainly in brain. Ketonuria detection by Rothera's test. Rothera's test. In starvation, we have hypoglycemia, lipolysis, and ketoacidosis, but not hypercholesterolemia. Question, important point. Try to understand. LCAT, do not miss. Free to ester form in the plasma under the guidance of HDL. Right, important point. Do not miss this. HMG-CoA synthase is the common enzyme between ketogenesis and cholesterol synthesis. What is the common enzyme, my dear friends, between glycogenesis and glycogenolysis? That is phosphoglucomutase. That is phosphoglucomutase, my dear friends. Please try to understand. Cholesterol can give rise to bile acid, bile salts, vitamin D and steroid hormone, but not except bile pigments. Except bile pigments. Lipoprotein chemistry and metabolism, my dear friends. Please try to understand. We have exogenous TAG that is carried by chylomicron. Endogenous TAG is carried by VLDL, right? Important point. Please try to understand. HDL, reverse cholesterol transport. Ideal ratio of LDL HDL should be less than 2 because more than 2 precipitates atherosclerosis. LDL is beta lipoprotein and HDL is alpha lipoprotein. Please try to understand. HDL, there is no ApoB. Chylomicron contains ApoB48. Only apoprotein present in LDL is ApoB100. ApoE4 Alzheimer's disease. Lipoprotein small a, little rascal, highly atherogenic. A1 is the activator of LCAT, but C2 is the activator of lipoprotein lipase. Right, lipoprotein lipase, where it is not present, liver. Liver does not have LPL. Where it is present maximum, heart. It is present maximum, my dear friends. Please try to understand, my dear friends. These things are important point. So during starvation, all fats are mobilized from adipose tissue to the heart. Please try to understand. We have Fredrickson-Levy-List classification where we have all the hyperlipoproteinemia. But that classification, my dear friends, Fredrickson-Levy-List classification, it does not include HDL. Yes, important question. Please try to understand. HDL. HDL is not included in those type 1, type 2A, type 2B, type 3, type 4 and type 5. None of the disorders talk about HDL. All are with others, either chylomicron, LDL, VLDL and IDL, but never ever HDL. HDL disorders are not included in this group of hyperlipoproteinemias. Yes, important. This is the question that has been asked recently. Which of the following is not included as per Fredrickson-Levy-List classification? Yes. Coming to type 2A, already marked in green. Do not miss this. Fish eye disease, you may remember familial LCAT deficiency. Tangiers disease, you may remember orange yellow tonsils. Nucleotide chemistry, I told you to remember gout. Do not miss gout, my dear friends. Gout, please do not miss. Monosodium lutate crystals. Try to understand this. Most common joint, first metatarsophalangeal joint. Leishman syndrome, do not miss. do not miss. So with this, we basically have seen the most important questions from the topics that we have discussed during our classes deliberately. So please remember, vitamins, we have already seen the questions. If you can stretch, you can go through these vitamins questions. And please try to understand molecular biology we are not touching. At this section, I have also given you all the recent years questions, my dear friends. Whenever you guys are free, you can just attempt these questions. These are your recent past years questions. Please go through them. Please go through them. Answers also has been provided to you so that you can take these things as a practice session. So please go through these questions. Do not miss these previous year's questions, my dear friends. That has been recently asked. What is this image? We have seen this image, galactosemia. Yes, yes, oil drop cataract. Like please go through these images, my dear friends. What is this image? We have seen this same image we have seen in the class. These are actually xanthomas over the extensor tendon of our hand, primary familial hypercholesterolemia, that is tendon xanthoma. So like this, many questions has been given. What is this image? Yes, just now we have seen. This is niacin deficiency pellagra, patchy dermatitis. Initially Casals necklace, then patchy dermatitis. So like this, what is this image? Ricket, wrist widening. See, wrist widening. Wrist widening is C in rickets. So try to go through these previous year's questions. I have given you many questions for which you can just go through. This will be really helpful. Answer key is provided at the end. So don't just look for the answer. First try to attempt. You mark it with a highlighter. Then you come back to this section and check for the answers, right, dear friends, right? Important point to be noted. And I have also given you one visuals, right, dear my dear friends. You may go through this corneal clouding, pot belly, doll-like faces, those Coca-Cola, myoglobinuria, then those Heinz bodies picture, Benedict test picture, snowflake cataract, sunflower cataract, galactosemia image, blue sclera image. This is the image of the triple helical structure of collagen. All these things we have discussed, my dear friends. Please try to understand. This is that albuminuria detection wala test. This is the stretchability of the skin, Ehlers-Danlos syndrome, all those. So these are the images also from the osteogenesis imperfecta. This is our phenylketonuria blonde, white hair albinism, alkaptonuria image clusters we can see, homocystinuria, lens dislocation, downward lens dislocation. These are zebra bodies in Niemann-Pick disease. These are cherry red spot in macula, cherry tree never grow tall in sand, crumpled, wrinkled tissue paper appearance of cytoplasm. This is seen in Gaucher's disease. Then we have eruptive type of xanthoma that is seen in type 1 hyperlipoproteinemia, milky fat accumulation over the collected blood sample type 1. And these are the images, my dear friends. These three, please to understand this. Try to understand, my dear. Xanthelasma, we have corneal arcus type 2A, Palmer xanthoma, it is seen in type 3, acanthocytes A beta lipoproteinemia. This is also, please try to understand, my dear friends. We have tendon xanthoma. Tendon xanthoma is again type 2A. These are monosodium iodide crystals. These are tubero-eruptive type of xanthoma. This is image based question, orange yellow tonsils. And we have also added, I have also added, my dear friends, few image based questions from the vitamin section as well. This guy that you are seeing from here, he is lethargic, he is weak. This is actually the picture of a patient of beriberi. This is a recently given image in the NEET PG exam, so they might give you this image as well, beriberi image. These two are the images of our Lish-Nihan syndrome, self mutilation. We have seen those. Please remember, vitamin B2 deficiency can present with angular stomatitis and cheilosis. Please remember, this is that you are seeing is also glossitis. This is not the geographic tongue. Geographic tongue is different than this. This is not the geographic tongue. Geographic tongue is like a map, it will be seeing. That is in different one. Here it is glossitis. This is circumcorneal vascularization. This is seborrheic dermatitis, different forms that can be seen again in vitamin B2 deficiency. Vitamin B2 deficiency. This is pellagra. This is also dermatitis, patch, rough skin over the hand. This is Gopalan's burning foot syndrome, pantothenic acid deficiency, sideroblastic anemia, B6 deficiency, petechial hemorrhages, vitamin C deficiency, macrocytic anemia, vitamin B9, megaloblastic anemia. We have hypersegmented neutrophils. This is classically seen in B12 deficiency. We have gum bleeding in scurvy. Important point. We have also some images from the vitamin A. We have xerophthalmia, Severe form is converted to keratomalacia. These are bite spots, triangular white colored plaques that can be given. Please remember, both of these that you are seeing can be seen in rickets. This is bow-leggedness valgus and this is valgus, that is we have this knock knee. This can both be seen in vitamin D deficiency ricket. This is also an image in ricket. This is known as Harrison's sulcus. If you try to ask the child to move like this, there will be a groove-like thing that is formed, that is known as Harrison's sulcus and rickety rosary. Beaded appearance of the ribs. Whenever you ask the child to stretch like this, groove and then beaded appearance, this is also called as rickety rosary, Harrison's sulcus. Please remember, this three image cluster that you are seeing over here, rashes over the genitalia, then also over the hands, also over the mouth. This is classically seen in acrodermatitis enteropathica, that is due to zinc. Yes, not vitamin. Everything is not vitamin. This is zinc deficiency, acrodermatitis enteropathica. And this gentleman, we have seen that he was suffering from some osteochondropathy changes, that is seen in Kashin-Beck disease, that is also due to deficiency of selenium. So with this, we basically finish the quick revision points of entire biochemistry, my dear friends. So as you can see that we have revised first time in just 2 hours time entire biochemistry. Yes, it can be done. If you have written down notes sincerely, you can do it in just 2 hours as well. So we have started, and the next time when we have done it hasn't even taken 1 hour for us. So when you are going through this video, maybe you have to watch this entire session of video for 2 times, then you are done. Believe me, then you are done. You will be definitely scoring more than 15 MCQ correct in your upcoming exam if you just go through this video, my dear friends. Please remember, at least watch this video 2 times. So it will be a revision for you for 4 times of biochemistry. In these 3 hours, we have seen previous year's questions targeted, image-based questions, as well as the main content that we have highlighted. From there also, next time we have highlighted in green color highlighter. Those are most important MCQ. So do not miss those, my dear friends. So wish you guys all the best. I have immense belief in all of you, my dear friends. I have immense belief in all of you. You just need to trust yourself. I trust in you guys. And I know you guys will be shining like stars on day 4th of December of this year. Please keep your morale high and always go with a positive mindset and positive attitude that yes, this is the day that I was waiting for, and this is the day, my dear friends, that you all will be marking the questions correctly. Not only from biochem, but from all the topics. Yes. So wish you all the best, my dear friends. Just the last stretch of few days. Give your best. Put more than 100% of your efforts. And believe me, my dear friends, success will be yours. So do contact me. As soon as you have written the exams, we will discuss the previous year's questions and the question that has come in your exams as well. So wish you all the best, my dear friends. And I know when the results are out, you all will be shining like stars. Right. This is what we want from the entire team of MIST, is your success, my dear friends. So thank you all. Wish you all the best. Ekdam 4 तारीख 4th December 2022. Jhanda gaad ke aana. Yes. Yes, that is the attitude, my dear friends. So all the best and wish you good luck. Thank you.